A fragile organ rests inside the skull, 3 pounds of tissue, yet capable of language, memory, and imagination. How can matter this soft hold the architecture of thought itself? Welcome to NeuroRest, where we explore the essentials of neuroscience in a calm, structured way. Today we'll travel from brainstem to cortex, tracing how circuits create sensation, memory, and decision. To make this journey complete, I've prepared a free practical guide with a concise summary of today's lesson, waiting in the description like a map you can return to whenever needed. Settle in. Let's begin with the living mystery of the brain. Inside your skull lies a fragile structure that feels soft, almost jelly-like, yet it is the seat of every thought, dream, and decision you have ever had. The brain is paradoxical in this way, a material organ weighing only a few pounds, yet capable of generating the richness of human experience. To open our exploration, we begin with its essence as a living organ of mystery. When scientists first examined the brain centuries ago, they were struck by its lack of obvious machinery. Unlike the heart with its chambers, or the lungs with their branching airways, the brain appeared to be a mass of uniform tissue. But with time, new methods revealed patterns hidden within. Under the microscope, neurons emerged as discrete units, each with a cell body, slender branches, and a long projecting fiber called an axon. These neurons do not operate alone. Each connects to thousands of others, forming networks with a density that almost defies imagination. The apparent uniformity of the brain gave way to an intricate architecture of circuits. This idea that order can emerge from seemingly formless tissue is central to neuroscience. The brain is not random mush but organized layers of cells with distinct roles. Gray matter houses dense clusters of cell bodies where processing occurs, while white matter carries signals across long distances. These two types of tissue cooperate seamlessly. You can picture gray matter as cities where messages are generated, and white matter as highways along which they travel. Even this simplified map already suggests how extraordinary coordination is possible. At the surface, the brain folds into ridges and valleys. These folds enlarge the surface area, allowing more cells to pack into the skull. Each major fold corresponds to a functional neighborhood. Some regions handle movement, others sensation, and still others abstract thought. When you speak, recall a face, or plan your day, those activities are not dispersed at random. They are rooted in specialized areas of cortex. The first seeds of modern neuroscience sprouted when physicians noticed that injuries in different locations produce different losses, language after one type of lesion, movement after another. That realization confirmed the principle of specialization. Another layer of mystery lies in the brain's dual nature. It is physical, bound by biology, yet it generates subjective experience. When you remember a childhood event, no one can observe that memory directly, but they can see the hippocampus and related networks working. This contrast between private inner life and public neural signals defines much of the scientific challenge. Researchers measure electrical impulses, blood flow, or chemical release, but what those measures feel like from the inside is something only the individual can report. Thus, brain science always moves between two perspectives, the objective and the subjective. Understanding the brain as living tissue also reveals its vulnerabilities. It demands a constant supply of oxygen and glucose, and even brief interruption can impair function. Head trauma, infections, or strokes remind us that this organ, though powerful, is delicate. The paradox deepens. The most advanced computational system we know is also among the most fragile, and yet resilience is also evident. Many people recover partial function after damage, and children's brains can reorganize in ways adult brains rarely match. This dual capacity, fragility alongside adaptability, adds to the sense of awe. Studying the brain as tissue further reminds us of its continuity with other animals. All vertebrates have nervous systems built from neurons and glia. In fish, birds, and mammals, we find variations on the same basic plan. But in humans, certain regions expand dramatically, especially the cortex, giving rise to symbolic language, complex planning, and culture. This continuity shows that the brain is not only a human organ, but also a product of evolutionary history. Its mysteries are written in a shared biological script, with each species offering clues about our own design. In reflecting on the brain as a living organ, we also confront limits of knowledge. Even with imaging and molecular tools, much remains opaque. How exactly a burst of electrical activity translates into the feeling of joy, or how networks of billions of cells sustain consciousness, are still open questions. Neuroscience progresses, but humility is essential. To hold the brain in mind is to recognize both what we know and the vastness of what we do not. As this first step closes, let the image settle, a soft, folded organ, sustained by blood, packed with cells, whispering in electric patterns, somehow giving rise to all you are. In the next part of our journey, we descend to the base of the brain and trace its climb upward, from the simplest survival functions to the loftiest human thoughts. At the base of the skull lies a region that sustains life at its most elemental level. This area, called the brainstem, connects the brain to the spinal cord. It is small in volume compared to the rest of the brain, yet it carries out functions so vital that even brief damage can be catastrophic. Here, networks regulate heartbeat, breathing, and reflexes. Unlike higher brain regions that you can voluntarily control, the brainstem operates automatically. You do not need to think about inhaling or adjusting blood pressure. These rhythms unfold beneath awareness, hour after hour. The brainstem is often described in layers. The lowest part, the medulla, governs breathing and heart rhythms. Above it, the pons serves as a bridge, linking the cerebellum with other brain structures. The midbrain sits higher still, coordinating eye movements, reflexive responses to sound and light, and the release of certain neurotransmitters. Together, these structures form the core that keeps the body alive and responsive. If the cortex is the orchestra performing a symphony, the brainstem is the conductor that ensures the instruments are tuned and time is kept. Ascending from the brainstem, functions gradually shift from reflexive to more deliberate. The thalamus, located just above, acts as a relay. It filters incoming sensory signals and routes them to the appropriate cortical regions. Nearly every sensation you perceive passes through the thalamus. This flow underscores a broader theme. The brain is organized hierarchically, with lower centers handling basic life support and higher centers enabling thought and reflection. The cerebellum, tucked beneath the back of the brain, also emerges from this lower level. Long thought to control only balance and coordination, it is now known to play roles in cognition and emotion as well. Its densely packed neurons help fine-tune movements and timing. When you walk across uneven ground or learn a new skill, the cerebellum is actively adjusting output so your motions are smooth. The journey upward highlights a principle: complexity builds upon stability. Without the brainstem, there would be no steady platform for awareness. Without the thalamus, sensory chaos would overwhelm perception. Each higher function relies on deeper, older systems. Evolution sculpted this architecture over millions of years, preserving ancient circuits and layering new capabilities on top. That history is still visible in anatomy. The brainstem resembles structures found in reptiles, while the expanded cortex of humans represents more recent innovation. Studying this vertical progression also reveals clinical insights. A stroke in the brainstem may disrupt breathing or swallowing. Injury to the cerebellum produces unsteady gait and slurred speech. By contrast, damage higher in the cortex affects language or memory. These differences confirm the principle of specialization across levels. They also remind us that while the cortex often receives attention for its role in thought, the lower regions sustain the possibility of thought itself. The base-to-top organization offers a useful metaphor. Imagine climbing a mountain. At the bottom, the essentials of survival are secured: air, circulation, and reflexive responses. As you ascend, the view broadens. Higher ground affords perception, memory, imagination, and self-awareness. Yet no climber can reach the summit without a firm base. Similarly, the highest reaches of human consciousness stand on the foundation of brainstem rhythms and thalamic relays. To contemplate this layered ascent is to feel both humility and wonder. The functions that seem most automatic, breathing, heartbeat, reflexes, are the very ones that sustain the platform on which our lives unfold. In this way, the base of the brain is not primitive in a pejorative sense, but fundamental in a structural one. It anchors every act of thought, creativity, and connection. As we pause, picture this climb. At the base, hidden yet steady, ancient circuits keep the body alive. Step by step, networks ascend toward regions that govern perception and choice. In the next section, we will narrow our focus further, exploring the smallest players in this system, the neurons that send electrical whispers from one cell to the next, the building blocks of all higher function. At the smallest scale of the nervous system lie neurons, the fundamental messengers of the brain. Each neuron is a living cell, but unlike most cells in the body, its shape is elongated and branching, designed for communication across distances. At one end, tree-like structures called dendrites receive incoming signals. At the other, a long fiber called an axon carries signals away. Between neurons, Connections form at junctions known as synapses. It is through this elaborate wiring that the brain speaks to itself. The electrical language of neurons begins with a difference in charge across the cell membrane. When conditions are right, the neuron suddenly fires an action potential, a rapid electrical impulse that races down the axon. At the end of the axon, this signal triggers the release of chemical messengers called neurotransmitters. These molecules cross the synaptic gap and bind to receptors on neighboring cells, opening channels and continuing the chain of activity. The process takes only milliseconds, yet it underlies every thought, perception, and memory. One striking feature is the sheer scale of connections. A single neuron may form thousands of synapses with others. This dense web means that no cell acts in isolation. Instead, patterns of activity across networks determine perception and behavior. Imagine standing in a crowd where voices call out in rapid succession. The meaning is not in one voice alone, but in the chorus. In a similar way, neurons collectively encode information by firing in coordinated patterns. Different neurotransmitters lend nuance to this system. Glutamate excites, pushing neurons to fire. GABA inhibits, quieting activity. Dopamine modulates reward and motivation, while serotonin influences mood and rhythm. Acetylcholine shapes attention and learning. These chemical differences give networks distinct personalities, allowing the brain to regulate emotion, decision, and movement in subtle ways. Another layer of complexity comes from plasticity, the ability of synapses to strengthen or weaken over time. When two neurons fire together repeatedly, their connection grows more efficient, a principle often summarized as cells that fire together, wire together. This adaptability is the biological basis of learning. Whether practicing a musical instrument or memorizing new vocabulary, changes at the synaptic level gradually embed new skills into the brain's fabric. Yet neurons alone cannot explain the full function of the brain. They require energy, maintenance, and support. Surrounding them are glial cells, long underestimated but now recognized as essential partners. Some glia insulate axons with myelin, speeding conduction. Others clear debris, regulate chemical balance, or guide growth. The dance between neurons and glia sustains the living circuits that make consciousness possible. Clinical observations highlight the stakes of this microscopic communication. Disorders of neurotransmission can alter mood, perception, and movement. For instance, when dopamine pathways falter, movement becomes rigid and slowed. When serotonin signaling changes, mood and sleep can shift. These examples reveal how delicate balances at the synapse ripple outward into the texture of daily life. Despite decades of study, many mysteries remain. How does a specific pattern of firing across millions of neurons create the sensation of a face, the memory of a childhood song, or the urge to speak? Researchers can map circuits and record spikes, yet the translation from electrical signals to subjective experience is still elusive. The puzzle draws scientists deeper into both biology and philosophy. As we close this section, hold the image of neurons as tiny sparks, each whispering to its neighbors in chemical codes. Their combined activity forms the foundation of every skill and memory. In the next part of our journey, we will turn to the silent allies of neurons, the glial cells, whose support shapes learning, repair, and resilience. While neurons carry the headlines of brain science, a quieter population of cells works constantly in the background. These are glial cells, often simply called glia. For many decades, they were dismissed as passive scaffolding, the glue that held neurons in place. Yet modern research reveals that glia are active players in brain health, learning, and recovery. They may not fire electrical impulses, but their influence on neural circuits is profound. There are several main types of glia, each with distinct duties. Astrocytes, shaped like stars, stretch their arms across multiple synapses. They regulate the chemical environment, mop up excess neurotransmitters, and release signals of their own that influence whether a connection strengthens or weakens. In this way, astrocytes help sculpt learning at the most intimate scale. Another type, oligodendrocytes, provides insulation. They wrap axons in layers of fatty membrane called myelin, which speeds the conduction of electrical impulses. A signal that would otherwise crawl along a fiber instead leaps forward in rapid bursts. This efficiency allows distant parts of the brain to coordinate quickly, enabling complex behaviors like language or fine motor control. Without this insulation, signals falter, and timing is disrupted. Microglia serve as the brain's immune system. They constantly patrol the tissue, pruning away damaged synapses, engulfing debris, and responding to infection. During development, microglia also shape circuits by trimming excess connections, leaving behind streamlined networks. In adulthood, they remain vigilant, adjusting activity to maintain balance. When microglia malfunction, inflammation can spread, and this has been linked to various neurological conditions. Glia also play roles in repair. When neurons are injured, certain glial cells migrate to the site, sealing wounds and attempting to restore balance. Although neurons themselves rarely divide after birth, glial cells retain more flexibility. They can multiply, adapt, and even guide the growth of new neural connections. This plastic quality makes them central to resilience after trauma. The partnership between neurons and glia is so close that it is difficult to speak of one without the other. Neurons may generate signals, but glia maintain the environment that makes signaling possible. Without glia, neurons would drown in chemical noise, starve for energy, or fail to transmit quickly enough. What once seemed like supporting actors are now recognized as co-stars in the drama of brain function. Research continues to reveal unexpected roles. Some studies suggest that astrocytes contribute to the regulation of sleep, releasing molecules that influence when networks fall silent. Others show that glia interact with blood vessels, adjusting blood flow to meet local demands. These findings suggest that glia help coordinate the supply lines of energy, ensuring that busy neural regions receive what they need. Considering glia also highlights limits of our current understanding. For centuries, focus on neurons overshadowed these cells. Only recently have tools allowed precise tracking of their activity. As knowledge grows, new questions arise. How do glia influence memory consolidation? Could they be targeted to enhance recovery after injury? Might they hold keys to neurodegenerative disease? Each discovery reshapes our view of what the brain truly is, a community of diverse cells, not merely a network of firing axons. To reflect on glia is to appreciate subtlety. Their work is often unseen, unfolding in silence, yet it supports the brilliance of neural signaling. Like stagehands in a theater, glia ensure the lights shine, the sound carries, and the stage is ready. Without them, the performance would collapse. As this section closes, picture the neurons as vibrant sparks, and the glia as the steady field that nurtures them. In the next chapter, we will move from individual cells to the level of circuits, exploring how collections of neurons and glia form repeating patterns that give rise to perception and action. When many neurons work together, they form circuits, organized pathways that repeat their patterns of activity in reliable ways. These circuits are the bridge between single-cell signals and the coherent functions we recognize as perception, memory, or movement. The concept of a circuit emphasizes that the brain is not random chatter but structured flow, where signals travel along defined routes. A simple example lies in reflex arcs. When you touch a hot surface, sensory neurons send a rapid signal to the spinal cord, which loops back through motor neurons to contract your muscles. This circuit bypasses conscious thought, ensuring immediate withdrawal. While basic, such reflexes illustrate the principle. Information flows through predictable loops to produce a specific outcome. In more complex circuits, signals pass through multiple regions. Consider vision. Light hitting the retina travels through the optic nerve to the thalamus, then onward to visual cortex. Within the cortex, specialized patches handle edges, colors, and motion. Each unit processes part of the picture, and their combined activity yields coherent sight. The path is not a single straight line but a web of parallel and feedback loops. Circuits allow these processes to run efficiently, so perception feels instantaneous. Another striking example is the basal ganglia, a set of interconnected nuclei that regulate movement and habit. Signals cycle through loops linking cortex, basal ganglia, and thalamus, refining actions. When these circuits malfunction, movement becomes slow or rigid, as seen in certain neurological conditions. These loops illustrate how circuits balance excitation and inhibition to fine-tune behavior. At the heart of circuits lies the concept of convergence and divergence. Convergence occurs when many neurons funnel signals to a smaller set, sharpening information. Divergence occurs when one neuron sends signals to many targets, spreading influence. Together, these arrangements create flexibility and resilience. Even if individual neurons falter, the circuit as a whole can often maintain function. Importantly, circuits are dynamic. They strengthen with repeated use and weaken when unused, echoing the principle of plasticity. Musicians, athletes, and language learners all rely on circuit reinforcement. Over time, once effortful actions become automatic because the circuit has been tuned. This adaptability underscores why practice leaves lasting traces in the nervous system. Studying circuits has required creative tools. Early neuroscientists relied on observing deficits after injury, inferring which circuits were disrupted. Later, electrical stimulation allowed mapping of motor and sensory loops. Modern imaging reveals coordinated activity across regions, highlighting circuits in action. With each new method, our understanding shifts from isolated areas to networks working in concert. The circuit perspective also reframes how we think about the mind. Thoughts are not the product of single spots lighting up, but of entire ensembles firing in patterns. A memory of a familiar voice, for example, engages circuits spanning auditory, emotional, and associative regions. The richness of experience emerges from their synchronization. Reflecting on circuits offers a calming recognition of order. What seems like chaos inside the brain is in fact organized flow, where countless signals braid into pathways that repeat and adapt. This order allows perception to remain stable, even as the world around us shifts. As we conclude this section, picture the brain not as a tangled mass, but as a city of circuits, each route connecting neighborhoods of function. In the next part of our journey, we will examine how these circuits map onto the surface of the cortex, where orderly representations of the body and senses are laid out like cartography on living tissue. If you lift the surface of the brain into view, you encounter the cortex, a sheet of gray matter folded into ridges and valleys. Despite its wrinkled appearance, the cortex is remarkably orderly. Within its expanse lie maps, organized representations of the body, the senses, and even abstract information. These maps demonstrate that the brain is not an undifferentiated mass, but a structured landscape. The clearest example comes from the motor and somatosensory cortices. Here, body parts are laid out in topographic order, a layout often depicted as the homunculus. In this distorted figure, the hands and lips occupy far larger territory than the back or legs, reflecting their fine control and sensitivity. When an electrode stimulates a spot in motor cortex, a specific muscle group twitches. When a touch is applied to the skin, the corresponding patch of sensory cortex lights up. These maps reveal a consistent principle. Space on the cortex is devoted in proportion to functional importance, not physical size. Similar mapping extends to other senses. In the visual cortex, cells are arranged retinotopically, mirroring the layout of the retina. Neighboring points in visual space are represented by neighboring neurons in cortex, preserving spatial relationships. This arrangement allows precise reconstruction of images. In the auditory cortex, organization follows frequency, lower tones at one end, higher tones at the other. Such tonotopy underlies the ability to distinguish pitch and melody. These cortical maps are not fixed at birth. They shift with experience, injury, and training. Musicians often show expanded representations of the fingers used to play instruments. Individuals who lose a limb may find neighboring areas of cortex invading the unused space, sometimes contributing to the sensation of a phantom limb. These changes illustrate the brain's capacity to redraw its own maps, guided by activity. The maps also extend beyond the physical senses. Certain areas specialize in faces, places, or words, forming functional landmarks. These patches are consistent across individuals, suggesting a blueprint shared by human brains. For example, the fusiform face area responds strongly to faces, while the parahippocampal place area favors scenes. Such specialization hints that the cortex has carved out territories for categories of information vital to survival and social life. Understanding cortical maps offers clinical insights. Surgeons rely on them to avoid disrupting critical areas during operations. Rehabilitation therapies often aim to encourage remapping, coaxing the cortex to allocate new territory to recovering functions. By tracking how maps expand or contract, clinicians can monitor progress and tailor interventions. Reflecting on these maps provides a sense of elegance. Rather than chaos, the cortex reveals cartography, ordered layouts that echo the body and environment. This organization explains how signals remain coherent across vast networks. It also offers reassurance that the brain is not only complex, but structured in ways we can discern and describe. As we pause here, hold the image of the cortex as a living atlas, each fold containing landmarks that correspond to sensation, movement, or recognition. In the next section, we will follow signals deeper into one of the most elaborate maps of all, the pathways that turn patterns of light into the experience of vision. Vision begins with light striking the retina, but the sense we experience is constructed in the brain. The cortex transforms raw signals into coherent images, assigning edges, colors, motion, and depth. This transformation illustrates how perception is not passive reception but active interpretation. From the retina, signals travel through the optic nerve to the thalamus, specifically the lateral geniculate nucleus. Here, information is sorted before being relayed to primary visual cortex, located at the back of the brain. In this region, cells are organized to respond to simple features, edges at certain angles, contrasts of light and dark. These basic detectors form the foundation of visual analysis. From this starting point, information flows along two main pathways. One, often called the ventral stream, moves downward and forward into the temporal lobe. This pathway specializes in identifying objects, what something is. The other, the dorsal stream, extends upward into parietal regions, handling spatial relationships where something is and how it moves. Together, these streams create a dual representation of the world, blending identity and location. Along these streams, complexity increases. Cells in one area might respond to combinations of edges, forming shapes. In higher regions, responses become tuned to objects, faces, or entire scenes. By the time signals reach advanced visual areas, the brain is encoding meaning, not just patterns of light. This hierarchy allows us to recognize a friend's face instantly, even under changing light or from different angles. Vision also depends on feedback. Higher areas send signals back to earlier ones, shaping perception. When you expect to see something, your brain primes certain circuits, making recognition faster. This interaction shows that vision is not a simple feed-forward process, but a dialogue across levels. It explains phenomena like visual illusions, where expectation bends perception. Importantly, vision is intertwined with action. The dorsal stream equips us to grasp objects, navigate obstacles, and coordinate movement. Without it, recognition might remain intact, yet reaching accurately would falter. This link underscores how perception is embedded in behavior. The brain does not simply build pictures, it builds guides for action. Clinical cases illuminate these principles. Damage to the ventral stream can cause visual agnosia, where objects are seen but not recognized. Injury to the dorsal stream may produce difficulties with spatial coordination. Such dissociations confirm the division of labor within visual circuits. Reflection on vision brings a sense of wonder. What seems immediate and effortless is in fact the work of countless cells firing in ordered cascades. Each glance engages a sophisticated architecture, tuned over evolutionary time to extract meaning from photons. As we close this section, imagine vision not as passive seeing, but as active construction. In the next stage of our journey, we will turn to hearing, another sense that begins with simple vibrations, yet blossoms into speech, music, and meaning. Hearing begins with vibrations in air, but the experience of sound emerges only when the brain interprets those vibrations. The auditory system transforms pressure waves into patterns that carry meaning. Speech, music, warning signals, and subtle emotional tones. Like vision, hearing is a process of construction. Sound enters the ear and sets the eardrum vibrating. Tiny bones in the middle ear amplify these vibrations and transmit them to the fluid-filled cochlea. Inside this spiral structure, rows of hair cells convert mechanical motion into electrical signals. Each hair cell is tuned to a particular frequency, so the cochlea acts like a natural spectrum analyzer. From this point onward, the auditory system represents sounds as patterns of neural activity rather than airwaves. Signals travel through the auditory nerve to the brainstem, where timing is compared across both ears. This comparison allows localization, knowing from which direction a sound arises. The information then passes to the thalamus, and onward to auditory cortex, nestled in the temporal lobe. Here, neurons are arranged in a map of frequencies, from low to high, creating a tonotopic layout that mirrors the cochlea. Beyond basic mapping, auditory cortex integrates complex features. Some regions are sensitive to the rhythm and pitch of music, others to the rapid changes in speech. This specialization allows humans to decode language at remarkable speed, distinguishing phonemes that occur in fractions of a second. For music, networks track melody, harmony, and timbre, weaving vibrations into aesthetic experience. The brain does not process sound in isolation. Auditory regions interact with areas for memory, attention, and emotion. A song can trigger vivid recall of past events because auditory circuits link tightly with the hippocampus. A sudden noise can spark fear because signals reach the amygdala even before conscious recognition. These connections demonstrate that hearing is not merely about detecting sound but about assigning significance. Plasticity shapes auditory maps as well. Musicians often show expanded representation for tones they practice. People who learn a second language in adulthood may struggle with unfamiliar sounds because their auditory cortex has become tuned to the phonemes of their first language. These adaptations illustrate how experience sculpts perception at the neural level. Clinical insights underscore the importance of auditory processing. Damage to auditory cortex can cause difficulties in recognizing sounds, even when hearing itself is intact. Disorders affecting brainstem circuits can impair localization, making it hard to track conversations in noisy environments. Cochlear implants, which bypass damaged hair cells and directly stimulate auditory nerves, highlight how faithfully the brain can adapt to artificial input. Reflection on hearing emphasizes how the brain transforms vibration into meaning. What begins as simple oscillations in air ends as language, music, and emotional resonance. The cortex does not simply record sound, it interprets, categorizes, and links it to memory and feeling. As this section closes, imagine the brain as a silent hall that comes alive when waves of air are translated into messages. In the next stage, we will move to the sense of touch, exploring how the body's surface becomes mapped onto cortex, guiding action and perception alike. Touch anchors us to the world. Every brush of fabric, every step across the ground, and every gesture of the hand is mediated by networks that register pressure, vibration, temperature, and pain. The sense of touch is not only about surface contact, it guides posture, movement, and awareness of the body in space. Signals begin in the skin, where specialized receptors detect different forms of stimulation. Some respond to light pressure, others to stretch, vibration, or warmth. These receptors send impulses along peripheral nerves toward the spinal cord. From there, pathways ascend to the thalamus, and then to somatosensory cortex, where an organized map of the body is maintained. This cortical map, or homunculus, ensures that sensations from each body part are represented in consistent locations. The distribution of this map reflects function rather than size. Hands and lips occupy large portions of cortex because of their sensitivity and dexterity. In contrast, the back receives little cortical space. This unequal representation explains why a pinprick on the fingertip feels sharper than one on the shoulder. The brain devotes resources where precision is most useful. Touch also provides continuous feedback for movement. As you grasp a cup, pressure receptors signal how firmly you are holding it. If the grip slips, reflexive circuits adjust muscle tension within fractions of a second. Without this sensory guidance, even simple tasks become clumsy. Patients who lose touch sensation through nerve damage often struggle to button clothing or walk steadily, despite having intact muscles. Another dimension is proprioception, the sense of body position. Receptors in muscles and joints inform the brain about limb placement and movement. This internal feedback combines with surface touch to maintain balance and coordination. Even with eyes closed, you can reach for your nose because proprioceptive signals continuously update the brain about where your arm is in space. Touch also carries emotional weight. Gentle contact can soothe stress, activate bonding circuits, and release calming neurochemicals. Pain, by contrast, warns of danger and demands protective action. Both pleasant and unpleasant sensations are integrated with emotional centers, shaping behavior. Thus, touch is not only mechanical input, but a medium of connection and meaning. Plasticity is evident here as well. People who learn fine motor skills, such as playing string instruments, show expanded cortical representation of the fingertips. In cases of injury, neighboring regions of cortex may invade unused territory, sometimes leading to sensations that feel misplaced, such as phantom limb experiences. These changes demonstrate how the brain continuously redraws its map of the body. Clinical cases highlight the importance of touch circuits. Lesions in somatosensory cortex can produce numbness or distorted perception in specific body parts. Damage to spinal pathways may block sensation below the injury, yet rehabilitation often leverages plasticity, training the brain to reinterpret input from surviving pathways. Reflecting on touch brings calm recognition of the body's constant dialogue with its environment. Each contact is registered, sorted, and integrated into a coherent sense of self in space. This dialogue is so seamless that we rarely notice it until it is disrupted. As we conclude this section, hold the image of the cortex as a living map, where every brush of skin has its place. In the next stage, we will turn to senses more ancient and primal, smell and taste, which tie the brain directly to memory and emotion. Smell and taste are often called the chemical senses, because they rely on molecules rather than light or vibration. Though sometimes overlooked compared to vision and hearing, these senses are ancient and deeply tied to survival, memory, and emotion. They remind us that the brain is not only a cognitive machine, but also a detector of the world's most elemental signals. Smell begins in the nasal cavity, where airborne molecules bind to receptors on sensory neurons. Each receptor is tuned to a range of chemical shapes. When activated, these neurons send signals directly to the olfactory bulb, a structure at the base of the brain. From there, information spreads to cortical areas, without first passing through the thalamus, an exception among senses. This direct pathway explains the raw, immediate quality of smells. Taste arises from receptors clustered in taste buds on the tongue and other surfaces of the mouth. These receptors respond to categories such as sweet, salty, sour, bitter, and umami. Signals travel through cranial nerves to the brainstem, then to the thalamus and gustatory cortex. Although taste has fewer categories than smell, it combines with olfaction to produce the full richness of flavor. A blocked nose during illness reminds us how much taste depends on smell. The connections of these senses reveal their emotional weight. Olfactory and gustatory pathways project strongly to the limbic system, particularly the amygdala and hippocampus. As a result, a faint scent can trigger vivid memories, or a bitter taste can evoke instinctive aversion. These circuits illustrate how closely survival mechanisms are linked to perception. Foods that nourish are often experienced as pleasant, while those that may harm are experienced as bitter or repulsive. Cultural and personal experiences further shape these senses. A spice cherished in one cuisine may seem overwhelming to someone unaccustomed. Through repeated exposure, preferences shift, and the brain learns to interpret complex mixtures as familiar and desirable. This adaptability shows that even primal senses are subject to learning and memory. Clinical cases highlight their significance. Loss of smell, known as anosmia, can diminish appetite, blunt emotional experience, and reduce safety by making it harder to detect smoke or gas. Changes in taste perception can accompany illness or medication, altering the pleasure of eating. These conditions remind us how central chemical senses are to daily life, even if they often escape conscious notice. Research continues to uncover surprising roles. Some evidence suggests that smell influences social interactions, guiding attraction or signaling illness. Taste receptors have been found not only in the mouth but also in the gut, where they help regulate digestion. Such findings expand our view of these senses beyond simple perception. Reflecting on smell and taste highlights their primal yet profound nature. They are gateways to memory, emotion, and survival, tying the inner world of the brain to the chemical landscape around us. Their directness and emotional resonance make them unlike any other sense. As we close this section, imagine the aroma of a familiar place, or the flavor of a cherished meal. How quickly such sensations transport you through memory and feeling. In the next part of our journey, we will turn to the hippocampus, the brain's archivist, which records and retrieves the stories of our lives. The hippocampus sits deep within the temporal lobe, a small curved structure shaped like a seahorse, from which it takes its name. Despite its modest size, it is essential for recording the events of daily life and for retrieving them later. Without it, experiences would slip away, leaving the present moment unanchored to the past. To appreciate its role, it helps to imagine the hippocampus as a kind of living archivist, continuously cataloging the flow of experience into the broader library of memory. Each day, countless sensations reach the brain: sounds, sights, touches, emotions. Most pass through without leaving a lasting trace, but some are selected for storage. The hippocampus does not work alone in this process. It collaborates with surrounding cortices that process details of context, spatial layout, and emotion. Together, these regions bind scattered elements into coherent episodes. You remember not only the words of a conversation, but also the room where it took place, the tone of voice, and the feeling it left behind. The hippocampus knits these threads into a memory that can later be retrieved in vivid form. One of the hippocampus's striking features is its sensitivity to place. In animals, certain neurons within the hippocampus fire whenever the animal occupies a particular location. These place cells form an internal map of the environment, updating as the animal moves. In humans, similar activity supports navigation and spatial memory. When you recall how to walk through your home in the dark, the hippocampus is reactivating these stored maps. This spatial role is so central that many researchers see the hippocampus as a navigation system for both physical space and mental experience, charting paths not only through rooms and streets but through the episodes of a lifetime. Clinical observations reveal the hippocampus's necessity. Patients who suffer damage to both hippocampi, whether from injury, stroke, or disease, often develop profound anterograde amnesia. They can carry on conversations, solve puzzles, and even learn motor skills, but they cannot form new memories of daily events. A meeting with a friend vanishes once attention shifts. The past before the injury may remain intact, but the future becomes unrecorded. This separation between old and new memories illustrates the hippocampus's role in consolidation, transferring recent experiences into long-term storage across the cortex. The process of consolidation is gradual. Initially, the hippocampus plays a central role in replaying experiences, binding together distributed networks. Over time, as reactivation continues during sleep and waking rest, memories become stabilized in cortical circuits, allowing them to be recalled without hippocampal mediation. This shift explains why very old memories can survive even if the hippocampus is damaged later in life. The archivist has already filed them away securely. Sleep is particularly important in this process. During deep stages, hippocampal neurons replay patterns of activity from the day, as if rehearsing. These replays may occur in compressed time, allowing many experiences to be reinforced overnight. Such nocturnal rehearsals strengthen synaptic connections in cortex, embedding memories more deeply. This is why a good night of sleep often improves recall and problem-solving. Without hippocampal replay, experiences remain fragile and easily forgotten. The hippocampus is not only about memory storage but also about imagination. When people are asked to imagine future events, planning a vacation, envisioning a conversation, hippocampal circuits activate. This suggests that the hippocampus recombines elements of past experiences to construct possible futures. In this way, memory and imagination are two sides of the same function, the ability to arrange details into coherent scenes. Without this ability, life would not only lack record of the past but also foresight into what might come. Emotion also shapes what the hippocampus records. Experiences with strong emotional charge, whether joy or fear, are often remembered more vividly. This occurs because the amygdala, closely connected to the hippocampus, signals the importance of emotionally laden events. Stress hormones released during arousal can enhance consolidation, ensuring that certain experiences are prioritized. This mechanism helps explain why people remember dramatic events with particular clarity. Yet it also means that traumatic experiences can leave intrusive and persistent memories. Another dimension is neurogenesis, the birth of new neurons, which occurs in the hippocampus, even in adulthood. This discovery surprised neuroscientists, as most regions of the brain do not generate new cells after development. In the hippocampus, however, new neurons are continuously integrated into circuits. Their exact role is still debated, but evidence suggests they contribute to flexibility in learning and in distinguishing between similar experiences. This capacity may allow adaptation to new environments and help prevent memories from overlapping. Reflecting on the hippocampus also reveals its vulnerability. It is particularly sensitive to lack of oxygen, toxins, and stress. Conditions such as Alzheimer's disease often affect the hippocampus early, producing difficulties with recent memory. Prolonged stress can reduce its volume, likely through the impact of stress hormones on neural growth. These vulnerabilities underline the delicate balance that sustains the archivist of the brain. Yet even with fragility, the hippocampus shows resilience. Training, enriched environments, and regular physical activity have been associated with healthier hippocampal function. While not all decline can be prevented, evidence suggests that stimulation and engagement support its networks. This aligns with broader principles of plasticity. Active use strengthens connections, while disuse allows them to fade. The hippocampus is also a site where personal identity meets biology. Each individual's store of memories, the events, places, and relationships accumulated across years, forms a narrative of self. Without the hippocampus, this narrative frays, leaving only fragments. Its role in shaping identity highlights why memory loss can feel like loss of self. The archivist does not merely record events, it binds them into a story that gives continuity to life. Consider how memory enriches even ordinary experiences. A walk through a neighborhood carries echoes of past strolls, familiar landmarks, and remembered conversations. Meeting a friend is colored by years of shared moments. Without the hippocampus, these layers would be stripped away, leaving the present unconnected. The brain would still perceive sights and sounds, but without context, life would become a series of isolated snapshots. The hippocampus also interacts with language and culture. Storytelling relies on recalling and sequencing events. Learning history or literature requires connecting disparate details into coherent wholes. At the neural level, hippocampal circuits support this binding function, enabling humans to transmit knowledge across generations. In this way, the archivist of individual life also supports the archive of collective culture. Despite advances, many mysteries remain. How exactly does a distributed pattern of hippocampal activity become the vivid replay of a memory? Why are some experiences forgotten quickly, while others persist for decades? How does neurogenesis contribute to creativity and resilience? These questions continue to drive research, underscoring that the hippocampus, though small, is endlessly rich in function. As we reflect on this organ, its paradox becomes clear. It is both fragile and resilient, vulnerable to injury yet capable of lifelong growth. It anchors identity by recording the past, yet it also liberates imagination by constructing the future. Its continuous activity stitches moments into a coherent fabric, allowing life to feel like a story rather than scattered instants. To hold the hippocampus in mind is to appreciate the brain's ability to weave memory, imagination, and identity into one stream. It reminds us that the self is not static, but a narrative unfolding through time, recorded and replayed by networks of living tissue. As we close this extended meditation, imagine the hippocampus as a quiet librarian, shelves extending infinitely, binding the raw flow of experience into the archive that defines who you are. In the next stage of our journey, we will move from memory to emotion, exploring how another small structure, the amygdala, colors those stored events with fear, desire, and significance. Deep within the temporal lobe, adjacent to the hippocampus, lies a small almond-shaped structure that profoundly influences how we experience the world. This structure, the amygdala, does not record memories with detail or sequence as the hippocampus does, but it infuses those memories with emotional tone. It is the brain's sentinel, constantly scanning for significance, gauging whether an event is safe, dangerous, rewarding, or threatening. Without the amygdala, perception would be flatter, less charged, and memory less vivid. The amygdala receives input from nearly every sensory system. Visual cues, sounds, smells, and even internal bodily states converge here, allowing rapid evaluation of their meaning. This convergence means the amygdala can assign emotional weight before full conscious analysis occurs. A sudden rustle in the dark may trigger a startle before you realize it is only the wind. This immediacy reflects the evolutionary priority of survival. Better to overreact and live than hesitate and perish. Although most famous for its role in fear, the amygdala is not limited to negative emotion. It also processes positive signals such as pleasure and reward. Its networks interact with dopamine pathways, amplifying the salience of events that bring joy, excitement, or social bonding. This dual role makes the amygdala less a fear center than an emotional relevance detector. It highlights what matters, whether to avoid harm or to seek opportunity. The amygdala's influence reaches into nearly every domain of behavior. It modulates attention, drawing resources toward emotionally charged stimuli. A face showing anger or joy captures your gaze faster than a neutral one, because the amygdala flags it as important. It shapes memory by signaling the hippocampus to prioritize emotionally significant events. That is why you may recall where you were during a dramatic news event, or why a childhood embarrassment remains vivid decades later. The amygdala acts like a spotlight operator, deciding which scenes deserve to be recorded in high resolution. Clinical studies illustrate these roles. Individuals with amygdala damage often show blunted responses to fear and difficulty, recognizing fear in others' expressions. They may approach strangers without caution, failing to detect social cues of danger. Conversely, an overactive amygdala has been linked to anxiety disorders, post-traumatic stress, and phobias, where neutral or minor stimuli trigger overwhelming alarm. These extremes demonstrate how balance in amygdala activity is essential for adaptive behavior. The amygdala is also crucial in social life. It helps interpret facial expressions, vocal tones, and body language, allowing us to infer the feelings of others. Without this capacity, interactions become confusing or detached. In this way, the amygdala supports empathy and connection, not only vigilance. Its role extends into trust, bonding, and even moral judgment, revealing that emotion and social cognition are deeply intertwined. Another striking feature is how rapidly the amygdala can respond. Signals from the eye or ear can reach it through a fast, direct pathway that bypasses detailed cortical processing. This low road delivers crude but quick information, enough to trigger a jump at a shadow before the slower high road through Cortex confirms it is harmless. This dual route balances speed with accuracy, ensuring survival while still allowing refinement. The amygdala does not act in isolation. It communicates with the prefrontal cortex, which can amplify or suppress its responses. In moments of calm reflection, the prefrontal cortex may downregulate fear, reminding you that a plane flight is statistically safe. Under stress, however, the amygdala can override rational thought, flooding the body with arousal. This interplay illustrates the tension between emotion and reason, a dialogue at the core of human experience. The body is enlisted in these processes through the amygdala's connections with hypothalamic and brainstem circuits. Activation can trigger the release of stress hormones, accelerate heart rate, and sharpen attention. These physiological shifts prepare the body for action, whether fight or flight. Importantly, such responses can also be harnessed for positive motivation. Heightened arousal before a performance or competition can sharpen focus and energy. Memory and imagination are deeply colored by amygdala activity. When recalling a joyful celebration or a frightening accident, the vividness often stems from the emotional charge imposed by amygdala-hippocampus interactions. In imagination too, the amygdala helps simulate the feelings of possible futures, influencing decision-making. This blending of memory, emotion, and foresight highlights how integral the amygdala is to the continuity of self. Emerging research reveals that the amygdala participates in more subtle functions as well. It appears to track unpredictability and ambiguity, adjusting vigilance when outcomes are uncertain. It contributes to learning by signaling when expectations are violated, a function essential for adapting to changing environments. These roles suggest the amygdala is not simply reactive but predictive, constantly updating assessments of what the future may hold. Developmentally, the amygdala matures earlier than prefrontal control regions, which may explain why children often display intense emotions and limited regulation. As connections between amygdala and prefrontal cortex strengthen during adolescence and adulthood, emotional responses become more balanced. Yet this balance is always dynamic, shifting with stress, fatigue, and experience. The amygdala's involvement in psychiatric conditions underscores its centrality. In anxiety disorders, hyperactivity creates persistent worry and exaggerated startle. In depression, altered amygdala responses to negative stimuli may reinforce rumination. In post-traumatic stress, traumatic cues trigger re-experiencing through amygdala-driven arousal. Treatments often aim to recalibrate these circuits, whether through therapy, medication, or emerging approaches like neurofeedback. Despite its reputation for driving fear, the amygdala also plays a role in resilience. Some individuals exposed to trauma show adaptive responses, in part because their amygdala prefrontal circuits allow flexible regulation. Training, supportive environments, and repeated safe exposure can shape these networks, teaching the amygdala to recalibrate its alarms. This adaptability reflects the brain's broader plasticity, offering hope even in the face of adversity. Cultural and social influences further modulate amygdala function. Experiences of trust, prejudice, and group identity can alter its responses. For example, faces of unfamiliar groups may initially trigger stronger amygdala activation, though positive interaction can reduce this effect. Such findings reveal how deeply culture and learning sculpt what the amygdala deems significant. Even within daily life, the amygdala shapes subtle choices. The foods you crave, the people you approach, the risks you avoid, all are colored by its evaluations. Far from being a primitive leftover, it remains central to navigating a complex world. It ensures that perception is not only accurate, but meaningful, guiding behavior with an undercurrent of feeling. Reflecting on the amygdala invites awareness of how emotion and reason are interwoven. It reminds us that fear, joy, and desire are not obstacles to rational thought, but integral to it, highlighting priorities and motivating action. Without the amygdala's influence, decisions might be technically correct, yet devoid of personal significance. Emotion is not opposed to intelligence, it is part of what makes intelligence human. As we close this extended exploration, imagine the amygdala as a watchful companion, always scanning, always weighing, sometimes too reactive, sometimes life-saving. It does not merely color memory, it ensures that life carries urgency and meaning. In the next stage of our journey, we will ascend from this sentinel of emotion to the expansive fields of the prefrontal cortex, where reasoning, planning, and self-control emerge to balance the amygdala's intensity. At the very front of the brain lies the prefrontal cortex, an expanse of tissue that sets humans apart from most other species in its scale and complexity. It is here that the raw impulses of survival, colored by the amygdala, meet the deliberation of thought, strategy, and restraint. If the hippocampus serves as archivist and the amygdala as sentinel, the prefrontal cortex is the planner and negotiator, coordinating goals across time and mediating between emotion and reason. The prefrontal cortex can be divided into several functional zones, though in practice they work as an integrated whole. The dorsolateral regions are heavily involved in working memory and executive control. They allow you to hold a phone number in mind long enough to dial it, or to keep track of the steps of a recipe while cooking. This ability to maintain and manipulate information over short intervals is essential for problem-solving, reasoning, and flexible behavior. Without it, attention drifts, and tasks dissolve into confusion. Another important area is the ventromedial and orbitofrontal cortex, which are closely connected to emotion and reward systems. These regions help evaluate the potential consequences of choices, weighing risks and benefits. They are the seat of value-based decision-making, integrating signals from the amygdala, striatum, and other regions to produce judgments that balance immediate pleasure with long-term goals. When functioning well, they allow wise choices, such as saving resources now for future gain, or resisting an unhealthy temptation. When damaged, however, individuals may act impulsively, unable to anticipate the negative outcomes of their behavior. The anterior cingulate cortex, sitting on the border between prefrontal and limbic regions, monitors conflict and error. It detects when actions deviate from goals, generating the sense that something is wrong and needs correction. This monitoring system is crucial for learning from mistakes and adjusting strategies. Without it, behavior becomes rigid or error-prone, unable to adapt to changing circumstances. Together, these regions form a network for executive function, the set of capacities that allow planning, inhibition, reasoning, and social control. They provide the means to imagine future states, compare them with current conditions, and choose paths toward desired outcomes. They also mediate self-regulation, the ability to pause before acting, to weigh options, and to consider the perspective of others. These functions are often taken for granted, yet they are the foundation of complex society, law, education, and culture. The prefrontal cortex matures late in development, continuing to refine its connections into the third decade of life. This extended trajectory explains why adolescents often show heightened impulsivity and emotional reactivity. Their amygdala responses may be fully mature, but the regulatory circuits of prefrontal regions are still under construction. As connections strengthen, individuals typically gain better control of impulses, greater foresight, and more stable personality. This developmental story highlights how biology and experience intertwine in shaping behavior. Clinical evidence illustrates the prefrontal cortex's importance. Damage to this region, whether from injury, stroke, or disease, can produce profound changes in personality and judgment. Famous cases such as that of Phineas Gage, who survived an accident that destroyed much of his frontal lobe, show how intellect can remain intact while social behavior and self-control deteriorate. Gage, once reliable and courteous, became impulsive, profane, and unable to plan effectively. His case continues to demonstrate how the prefrontal cortex underlies the integration of intellect with social and moral responsibility. Modern neuroimaging confirms these functions. Tasks requiring reasoning, inhibition, or moral judgment consistently engage prefrontal circuits. When individuals suppress a habitual response in favor of a new one, dorsolateral areas activate. When they weigh ethical dilemmas, ventromedial regions show activity. These findings reveal that the prefrontal cortex is not a single-purpose area, but a hub for integrating information from across the brain, applying rules, goals, and values to guide action. Emotion and prefrontal function are deeply entwined. While the amygdala generates rapid evaluations, prefrontal regions can reframe them. This process, known as cognitive reappraisal, allows a person to reinterpret a threatening situation as manageable, or to view frustration as an opportunity for growth. Neuroimaging shows that when people successfully reappraise, prefrontal activity increases and amygdala responses decrease. This interplay underlies resilience and the capacity for emotional regulation. Another critical function is theory of mind, the ability to consider the beliefs and intentions of others. Prefrontal circuits, particularly in medial regions, support this capacity, allowing empathy, cooperation, and social negotiation. Without it, social interactions become shallow or confused. Disorders that impair prefrontal function, such as certain forms of autism or frontotemporal dementia, often disrupt social understanding, leading to difficulties in communication and relationships. Working memory, one of the hallmarks of prefrontal function, can be visualized as a mental workspace. Neurons in dorsolateral cortex sustain activity even when stimuli are no longer present, maintaining representations online for manipulation. This sustained firing allows the comparison of options, the sequencing of steps, and the planning of strategies. It is limited in capacity, which is why multitasking quickly overloads attention. Yet even within its limits, working memory provides a bridge between perception and decision. keeping information active until choices can be made. The prefrontal cortex also plays a role in creativity and abstract thought. It supports the combination of ideas, the generation of novel solutions, and the exploration of hypothetical scenarios. These functions depend on the integration of memory, imagination, and rule-based reasoning. The ability to consider not only what is but what could be distinguishes human cognition, and prefrontal circuits are central to that imaginative leap. Despite its sophistication, the prefrontal cortex is not infallible. It is highly sensitive to fatigue, stress, and intoxication. Sleep deprivation can impair its control, leading to impulsive decisions and difficulty concentrating. Stress hormones released during chronic tension can weaken prefrontal regulation, while strengthening amygdala reactivity, tipping the balance toward anxiety and rash action. These vulnerabilities highlight the need for balance and recovery in sustaining executive function. Plasticity within the prefrontal cortex means that training and practice can enhance its capacities. Mindfulness meditation, for example, has been shown to strengthen prefrontal regulation of attention and emotion. Cognitive training tasks can improve working memory and reasoning, though benefits often remain specific to practice skills. Education, social interaction, and problem-solving all serve as natural exercises for prefrontal circuits, sculpting their networks across the lifespan. The prefrontal cortex is also central to moral and ethical reasoning. Studies show that when individuals deliberate about fairness, harm, or justice, prefrontal regions become active. Damage here can lead to choices that prioritize personal gain over moral concern, even when individuals understand rules intellectually. This suggests that morality is not merely abstract reasoning, but an integration of values with decision-making circuits. The prefrontal cortex provides the stage on which moral intuitions, social norms, and logical reasoning meet. In reflecting on the prefrontal cortex, it is helpful to see it not as a commander, but as a negotiator. It does not silence the amygdala, hippocampus, or other regions, but coordinates them. It balances emotion with reason, short-term impulses with long-term goals, self-interest with social responsibility. It orchestrates rather than dictates, maintaining flexibility in the face of changing circumstances. The significance of this region for identity is profound. It supports the sense of continuity across time, the ability to plan for the future, and the capacity to regulate impulses in line with values. Without it, intellect may remain, but personality shifts, relationships fracture, and society struggles. Its health is therefore not only a matter of individual cognition, but of collective functioning. In daily life, the prefrontal cortex is at work in countless small ways. It helps you resist checking your phone during a conversation, guiding attention to the person before you. It allows you to budget, to prepare meals for the week, to weigh whether to speak or stay silent. It lets you imagine how your words will affect others, and to adjust accordingly. Each of these seemingly ordinary acts reflects complex orchestration of attention, memory, and emotional regulation. To contemplate the prefrontal cortex is to recognize the extraordinary capacity of the brain to integrate across domains. It reminds us that reason and emotion are not enemies but partners, that foresight and empathy are intertwined. It is the arena where humanity's greatest strengths, creativity, morality, sociality take shape. As this extended section draws to a close, imagine the prefrontal cortex as a wide terrace overlooking the rest of the brain. From here, one can survey memory, emotion, and sensation, weighing them in light of goals and values. The terrace does not control every detail below, but it provides perspective and guidance. In the next stage of our journey, we will explore how networks across the brain communicate, linking prefrontal oversight with deeper structures to create the unity of consciousness and action. The brain is not a collection of isolated modules but a web of networks, interlaced and dynamic, weaving perception, memory, emotion, and action into a seamless flow. Each region, from the hippocampus to the prefrontal cortex, plays its role, but it is their communication that produces the richness of consciousness and the fluidity of behavior. The study of these networks reveals how the brain achieves unity despite its complexity, and how balance across them supports adaptation to the changing demands of life. At rest, when the mind drifts without deliberate task, a constellation of regions called the default mode network becomes active. This network spans medial prefrontal cortex, posterior cingulate, precuneus, and parts of the temporal lobe. Its activity correlates with daydreaming, self-reflection, and the spontaneous retrieval of memory. It is the stage for inner narratives, imagining the past, and anticipating the future. When you catch yourself wandering through remembered conversations or planning tomorrow's schedule, the default mode network is at work, rehearsing possibilities and maintaining a sense of continuity of self. In contrast, when the mind engages with external tasks requiring focus, the executive control network rises in prominence. Anchored in dorsolateral prefrontal and parietal cortices, it sustains attention, suppresses distractions, and coordinates steps toward goals. The balance between this network and the default mode system is a kind of mental rhythm, shifting according to need. Too much inward focus and the external world is neglected. Too much task focus and reflection suffers. Healthy cognition involves the fluid transition between these states, orchestrated by hubs that regulate network switching. Another key network is the salience system, centered in anterior insula and anterior cingulate. It functions as a detector of importance, monitoring both the external environment and internal body states. When a stimulus is judged significant, a sudden sound, a shift in heart rate, or a novel idea, the salience network helps recruit attention and coordinate responses. In doing so, it acts as a mediator between default and executive systems, guiding whether to turn inward, outward, or toward immediate action. This network exemplifies how the brain prioritizes, ensuring that limited resources are directed where they matter most. The sensory and motor networks form further foundations. Visual, auditory, and somatosensory systems are linked not only to primary cortices, but to higher-order association areas that integrate across senses. Motor networks span from prefrontal planning regions to premotor and primary motor cortices, down through basal ganglia and cerebellum, ensuring that intention translates into precise action. These networks are bidirectional. Movement alters perception, and perception guides movement, in loops of feedback that create coherence between body and world. The hippocampal prefrontal network illustrates how memory and executive function collaborate. During decision-making, prefrontal circuits call upon hippocampal representations of past experiences, using them to model possible futures. This dialogue supports learning from history and anticipating outcomes. Functional imaging shows synchronized activity between these regions during planning and problem-solving, as if memory and foresight are two instruments playing in harmony. Disruption of their communication can impair the ability to use past lessons to guide present choices. Emotion networks further enrich this picture. The amygdala, ventral striatum, and orbitofrontal cortex interact in circuits of reward and threat. They link with brainstem systems that regulate physiological arousal, ensuring that emotion influences not only thought but bodily state. These circuits do not act alone but are embedded within larger networks. For example, when anticipating a rewarding event, amygdala activity couples with prefrontal oversight, balancing enthusiasm with judgment. When processing social cues, emotion networks align with theory of mind circuits, allowing feelings and empathy to converge. Communication across networks relies on both structural connections and rhythmic synchrony. The white matter tracts of the brain, bundled into highways such as the corpus callosum and arcuate fasciculus, provide the scaffolding for information transfer. But equally important are oscillations, patterns of rhythmic firing that coordinate distant regions. Just as musicians in an orchestra rely on rhythm to play together, neural populations use oscillations in different frequency bands to align activity across space. These rhythms allow signals to arrive in phase, enhancing communication efficiency. Disorders of brain function often involve disruptions of networks rather than isolated lesions. In schizophrenia, connectivity within default mode and executive networks is altered, contributing to disturbances of thought and self-experience. In depression, hyperconnectivity of default mode regions may underpin rumination, while weakened connectivity to executive systems impairs the ability to disengage from negative thoughts. In dementia, degradation of structural tracts disrupts coordination, leading to fragmentation of cognition. These observations shift the focus from individual areas to the integrity of communication among them. The study of networks also reframes plasticity. Training and experience not only sculpt local circuits but also reshape connectivity between regions. Learning a new language strengthens links between auditory, motor, and prefrontal areas. Meditation practices alter connectivity between default mode and salience networks, fostering greater awareness of mental states. Even recovery after injury often involves reorganization of networks, as surviving regions assume new roles or forge compensatory pathways. Reflection on brain networks invites reconsideration of consciousness itself. How does the subjective unity of experience arise from distributed activity? One hypothesis is that consciousness emerges from global workspace dynamics, where information is broadcast across networks through synchronized activity. Another emphasizes integration and differentiation. Consciousness requires both widespread sharing and local specialization. While debates continue, it is clear that the seamlessness of experience depends on the brain's capacity to weave together its diverse systems. In daily life, the fluidity of networks becomes evident in subtle shifts of attention and mood. A walk through a park may begin with executive control guiding footsteps, shift into default mode reflection about past events, be interrupted by salience detection of a bird's sudden call, and then return to inward planning. These transitions occur without effort, a testament to the brain's ability to manage multiple streams of activity. Each moment is the product not of a single area but of countless circuits working in concert. Social interaction highlights the importance of networks even further. Engaging in conversation requires auditory processing, language networks, theory of mind regions, and emotion circuits, all coordinated by prefrontal oversight. The ease with which humans exchange stories and ideas conceals the vast orchestration underneath. When this orchestration falters, as in certain neurological or psychiatric conditions, the difficulty of conversation reveals the complexity of what is usually effortless. Technological advances now allow direct observation of these networks in action. Functional MRI tracks blood flow correlations across regions, mapping connectivity patterns. Magnetoencephalography and electroencephalography reveal oscillatory synchrony. Diffusion imaging traces structural tracks. These tools provide windows into the living network, revealing how patterns change with learning, disease, or rest. They confirm that the brain is best understood not as a collection of nodes but as an interconnected system. Philosophically, the concept of networks challenges the idea of a central controller. Instead of a homunculus directing activity, coordination arises from distributed interactions. No single area contains the self. Rather, selfhood emerges from the interplay of memory, attention, emotion, and social cognition across networks. This perspective aligns with experiences of fragmentation under certain conditions, when disconnection leads to altered states of consciousness. It also aligns with resilience, when flexible reorganization maintains function despite loss. As with earlier reflections on individual structures, networks invite both humility and wonder. The brain's capacity to generate unity from complexity, to balance specialization with integration, reflects a principle seen across nature: the power of interconnected systems. Just as ecosystems rely on the interplay of species, or societies on the coordination of individuals, the brain thrives on the collaboration of networks. Its elegance lies not in isolation, but in connection. To close this extended meditation, picture the brain not as a static diagram of labeled parts, but as a living city at night. Lights flicker in neighborhoods, highways hum with traffic, signals cross bridges and tunnels. At times, districts coordinate for collective events. At other times, they rest in their own rhythms. Yet the city remains whole, vibrant, and adaptive. In the next stage of our journey, we will descend into the basal ganglia and cerebellum, structures often overlooked but vital for shaping movement, habit, and the rhythm of action that underlies the flow of thought itself. Beneath the sweeping folds of cortex lie structures often less celebrated in popular accounts of the brain, yet no less essential for the rhythm of thought and action. Among these are the basal ganglia and the cerebellum, both buried deep but shaping how intentions become movements, how repeated behaviors solidify into habits, and how coordination lends grace to even the simplest act. To reflect on them is to understand how the hidden machinery of the brain sustains both fluid motion and steady focus. The basal ganglia form a set of interconnected nuclei, nestled near the base of the forebrain. They include the caudate, putamen, globus pallidus, subthalamic nucleus, and substantia nigra. Though compact, they exert wide influence through loops that connect with cortex, thalamus, and brainstem. Their role is not to generate movement directly, but to regulate it, selecting useful actions and suppressing competing ones. This selection process underlies both physical and cognitive behavior, ensuring that at any moment the body and mind pursue coherent goals rather than scattered impulses. Signals flow into the basal ganglia from much of the cortex. They are processed through circuits often described as direct and indirect pathways. The direct pathway facilitates desired actions, while the indirect suppresses alternatives. Dopamine, released from neurons in the substantia nigra, modulates the balance between these routes. When dopamine levels are healthy, the system promotes smooth initiation of movement and flexible choice. When dopamine dwindles, as in Parkinson's disease, the system falters, producing difficulty in starting movement, slowed speech, and rigidity. This illustrates how a chemical imbalance in a small set of neurons can ripple across networks, altering the rhythm of daily life. Yet the basal ganglia are not limited to movement. They contribute to motivation, reward learning, and habit formation. Their loops with prefrontal and limbic regions help evaluate which actions are worth repeating, gradually automating them into routines. Driving a familiar route or typing without conscious effort depends on such circuitry. This automation conserves cognitive resources, freeing attention for novel challenges, but it also explains why breaking unwanted habits can be difficult. The basal ganglia have etched patterns into neural circuits that persist even when conscious goals shift. The cerebellum, situated at the back of the brain beneath the occipital lobes, is equally remarkable. Often called the little brain, it contains more neurons than the rest of the brain combined, densely packed into fine-grained circuits. Traditionally associated with balance and coordination, its role extends to timing, prediction, and even aspects of cognition. The cerebellum refines movements by comparing intended actions with sensory feedback, adjusting output to minimize error. This feedback loop operates with extraordinary speed, allowing fluidity in walking, speaking, or playing an instrument. When the cerebellum is damaged, movements become clumsy and uncoordinated. Speech may lose its rhythm, gestures may overshoot or tremble. Yet what stands out is not only the loss of grace, but also the disruption of timing. Patients struggle to judge intervals, clap in rhythm, or coordinate rapid sequences. These deficits suggest that the cerebellum functions as a predictive clock, aligning perception and action in time. Without it, behavior fragments, as if the music of life has lost its tempo. Research now reveals that cerebellar circuits extend into cognition and emotion. Connections with prefrontal cortex suggest roles in planning and working memory, while links with limbic regions imply contributions to effective regulation. People with cerebellar damage sometimes show changes in personality, difficulty with abstract reasoning, or blunted affect. These observations broaden our view, showing that the cerebellum is not merely a motor structure but a contributor to the rhythm of thought itself. Together, the basal ganglia and cerebellum illustrate complementary strategies of control. The basal ganglia act as gatekeepers, deciding which actions to initiate and which to inhibit, shaping behavior through reinforcement and habit. The cerebellum acts as a fine-tuner, correcting errors and refining performance through prediction and timing. Their cooperation allows behavior that is both purposeful and graceful, efficient yet adaptable. Consider the simple act of reaching for a glass of water. The prefrontal cortex sets the goal, the motor cortex generates the plan, but the basal ganglia help select the movement sequence while suppressing irrelevant gestures. As the hand moves, the cerebellum continuously adjusts trajectory, ensuring smoothness and accuracy. If the glass slips, basal ganglia circuits may trigger the habitual adjustment of grip, while cerebellar circuits fine-tune the correction in real time. Such collaboration makes complex actions seem effortless, though they are orchestrated by vast neural interplay. These systems also shape learning. When a novice practices a piano scale, basal ganglia circuits reinforce successful sequences through dopamine signals, gradually automating them into habit. Meanwhile, the cerebellum fine-tunes finger timing and coordination, reducing errors with each repetition. Over time, the combined work of both systems transforms effortful practice into fluent performance. This partnership illustrates how learning is not the work of a single brain region, but the symphony of many, each contributing a distinct instrument. Clinical conditions further highlight their importance. Parkinson's disease, rooted in dopamine depletion of the basal ganglia, produces motor slowing, but also affects mood and cognition. Huntington's disease, involving degeneration of basal ganglia circuits, leads to uncontrolled movements and psychiatric changes. Cerebellar ataxias disrupt coordination and balance, while developmental differences in cerebellar structure have been linked to autism, though mechanisms remain under investigation. These disorders remind us that beneath the surface of daily grace lies delicate circuitry, vulnerable yet vital. The basal ganglia and cerebellum also influence attention and thought. Basal ganglia circuits help filter mental actions, selecting relevant thoughts while suppressing distractions. The cerebellum, through predictive processing, may support the timing of speech and language comprehension. This cognitive reach demonstrates that even structures historically labeled motor contribute to the texture of mental life. They highlight the inseparability of movement, thought, and emotion. Habit formation, mediated by basal ganglia loops, illustrates how repeated behaviors carve grooves in neural pathways. With repetition, conscious effort fades, replaced by automatic execution. Habits can be beneficial, as in brushing teeth or exercising regularly, or detrimental, as in compulsions and addictions. Understanding the neural basis of habits offers strategies for change, introducing new cues, rewarding alternative behaviors, or deliberately engaging prefrontal oversight to override entrenched patterns. Yet the basal ganglia's efficiency means change is rarely instantaneous. It requires sustained practice to reshape circuits. The cerebellum's contribution to prediction extends beyond movement. When listening to speech, it helps anticipate syllables, smoothing comprehension. When following a melody, it predicts rhythm, aligning perception with expectation. This predictive function supports the brain's broader strategy to minimize surprise by continuously comparing predictions with input. In this sense, the cerebellum exemplifies a principle that spans the brain. Perception itself is guided by expectation, and action is shaped by forecast. Reflecting on these hidden structures deepens appreciation of the brain's architecture. They remind us that grace in movement and clarity in thought are not automatic, but the result of intricate regulation. They show that beneath conscious will lie mechanisms that sculpt, refine, and sustain behavior. Their contribution is not dramatic in isolation, but becomes indispensable in the flow of life. As we close this extended reflection, imagine the basal ganglia as the silent judge, weighing options and granting passage to some while withholding others. Imagine the cerebellum as the careful conductor, keeping time, correcting errors, ensuring harmony. Together, they allow the symphony of the mind to play with coherence and beauty. In the next stage of our journey, we will turn to the senses of time and rhythm themselves, exploring how the brain perceives duration, anticipates sequences, and binds experience into the continuous stream we call the present. Time is not something the brain perceives directly like light or sound. There are no receptors tuned to minutes or hours, no dedicated organ for measuring duration. Yet our experience is inseparable from time. Every movement requires timing. Every memory has a place in sequence, and every expectation depends on anticipating what comes next. The brain does not passively receive time, it constructs it, weaving rhythms, intervals, and continuity into the fabric of awareness. At the most basic level, neurons themselves have intrinsic rhythms. They fire in patterns shaped by ion flows and synaptic dynamics. These oscillations, ranging from slow cycles over many seconds to rapid bursts in milliseconds, provide a foundation for temporal coding. Networks synchronize their rhythms to align activity, allowing distributed regions to cooperate. When oscillations across areas fall into phase, information flows more efficiently. When they drift out of sync, coordination falters. This rhythmicity is one of the brain's tools for measuring and structuring time. Short intervals, on the scale of milliseconds to seconds, are crucial for speech, music, and coordinated action. The cerebellum plays a central role here, predicting the timing of sensory inputs and motor outputs. Without such prediction, speech would dissolve into noise, and music would lose its rhythm. Experiments show that people with cerebellar damage struggle to clap in time, or judge short durations, suggesting that this structure functions as a temporal calibrator. The basal ganglia also contribute, especially for intervals on the scale of seconds to minutes. Dopaminergic signals within these circuits appear to regulate internal clocks, speeding perception of time under some conditions, and slowing it under others. When dopamine levels are high, events may seem to pass more quickly, a phenomenon observed in states of excitement or stimulant use. When dopamine levels fall, as in Parkinson's disease, subjective time often drags, reflecting slower internal pacing. Beyond these subcortical contributions, the prefrontal cortex integrates temporal information with goals and memory. It allows us to hold a sense of duration in working memory, compare intervals, and estimate future delays. This function supports patience and planning. Without it, waiting in line or pacing oneself through study becomes intolerable. The prefrontal cortex thus links raw timing mechanisms to the psychological experience of duration. Time is also constructed through memory. The hippocampus not only encodes events but also sequences them, preserving their order. Place cells and time cells fire in patterns that mark not just where, but when events occur. This sequencing underlies the ability to tell stories, recall journeys, or anticipate the next step in a familiar routine. When hippocampal function is impaired, memories may lose their temporal context, becoming fragments without clear order. Our sense of the present moment depends on integrating information across short intervals. Visual perception, for instance, does not register each snapshot instantly, but blends inputs over hundreds of milliseconds. This creates continuity, smoothing rapid eye movements into stable experience. The brain thus constructs a specious present, a window of integration that feels like the now. Too short a window, and perception would fragment. Too long, and the present would blur into past. Emotion colors time as vividly as clocks measure it. Fear can stretch seconds into eternity, as heightened arousal accelerates internal pacing. Joyful absorption can collapse hours into moments, as attention narrows and self-awareness fades. These distortions arise from interactions between amygdala, basal ganglia, and prefrontal circuits, showing that time is not merely physical, but experiential. Subjective time flows according to the brain's evaluation of significance and engagement. Cultural practices reflect these neural underpinnings. Music organizes time into rhythm and meter, allowing groups to synchronize their movements and emotions. Rituals and ceremonies often rely on coordinated timing, reinforcing social cohesion. Even language encodes time metaphorically, shaping thought. Whether we see the future as ahead of us or approaching from behind depends on cultural convention. These expressions of time are grounded in neural circuits, yet expressed in shared meaning. Disorders of time perception reveal its fragility. Individuals with schizophrenia may experience difficulty sequencing events, leading to disjointed thought and disrupted sense of continuity. In depression, time may feel slowed, days dragging under the weight of low motivation. In attention deficit conditions, difficulty estimating intervals contributes to challenges in planning and sustained effort. These alterations remind us that time perception is not fixed but shaped by brain states. Modern neuroscience seeks to understand how distributed mechanisms converge into unified temporal experience. Some theories propose that time emerges from population coding, where patterns of activity across neurons represent duration. Others suggest that cascades of activity, like ripples spreading in a pond, provide markers of passage. Still others emphasize oscillatory hierarchies, where slow rhythms structure longer intervals and fast rhythms subdivide them. Whatever the mechanism, the result is a brain able to weave moments into sequence and sequence into narrative. Time also underlies anticipation. To catch a ball, the brain must predict its trajectory, estimating where and when it will land. To speak in conversation, it must time responses to the rhythm of speech. Anticipation depends on neural circuits that simulate the future, aligning action with unfolding events. The cerebellum, basal ganglia, and prefrontal cortex all contribute, each at different scales, ensuring that behavior is synchronized with the environment. Reflection on time reveals how deeply it structures identity. Memory anchors us in the past, imagination projects us into the future, and attention sustains the present. Together they create the sense of continuity we call self. When memory falters, as in dementia, continuity erodes, and with it the sense of identity. When imagination is curtailed, as in depression, the future feels closed. The construction of time is therefore inseparable from the construction of personhood. At the cellular level, even circadian rhythms embody temporal organization. So, prochiasmatic nuclei in the hypothalamus serve as master clocks, coordinating daily cycles of sleep, hormone release, and body temperature. These rhythms align with environmental cues like light, anchoring biological time to planetary time. Disruption, as in jet lag or shift work, strains cognition and health, underscoring how deeply our biology is tuned to cycles. Philosophically, the brain's construction of time raises questions about reality. If time is experienced depends on neural mechanisms, then the flow of moments is partly a creation of the mind. Physics describes time as dimension, symmetrical in equations, yet experience insists on direction, from past to future. The arrow of time we feel may emerge from memory and anticipation, functions deeply rooted in hippocampal and cortical networks. In this sense, the brain not only perceives but enacts time. Daily life illustrates these principles constantly. A child impatiently awaiting a holiday feels days stretch endlessly, while an adult engrossed in work finds weeks vanish swiftly. Athletes train to synchronize movements with fractions of seconds, while musicians align their phrases across beats. Each activity relies on circuits that measure, predict, and sculpt time, making it not abstract but practical, embodied in action. The adaptability of time perception shows the brain's flexibility. Under meditation, practitioners often report altered sense of duration, minutes feeling expansive or collapsing. In flow states, concentration so deep that action seems effortless, time may appear suspended. These experiences reflect shifts in balance among attention, memory, and emotion networks, highlighting how plastic and context-dependent temporal awareness can be. To appreciate the brain's construction of time is to recognize that every moment we feel is a synthesis. Neurons oscillate, networks synchronize, memories anchor, predictions project, and emotions stretch or compress. Out of these processes emerges the flow of now, the continuity of before, and the anticipation of after. The mind's narrative, the sense of self moving through life, rests on this hidden architecture. As we close this extended exploration, picture the brain as both clock and storyteller, ticking in rhythms yet weaving them into meaning. Time is not simply measured, it is lived, felt, stretched, and compressed. It is the medium through which consciousness unfolds. In the next stage of our journey, we will turn to language, the uniquely human capacity that allows us to encode time in words, to tell stories that span generations, and to share the inner flow of experience with others. Language is one of the most distinctive achievements of the human brain, a system that transforms thought into sound, gesture, and script, allowing ideas to cross from one mind to another. It is not merely a tool for communication, but a structure that shapes how we perceive, remember, and imagine. Through language, abstract concepts are named, stories are told, and cultures are built. To explore how the brain creates language is to glimpse the machinery behind meaning itself. The journey of language in the brain begins with perception. Sounds reach the ear as waves of pressure, converted into electrical signals by the cochlea, and then relayed to auditory cortex. There, in the superior temporal gyrus, neurons respond to features of speech such as pitch, rhythm, and phonemes. Yet perception alone is not enough. The brain must parse streams of sound into recognizable units. Specialized regions within the left hemisphere, particularly in the posterior superior temporal sulcus, excel at segmenting speech, distinguishing meaningful syllables from background noise. From perception arises comprehension. The classic territory known as Wernicke's area, located in the posterior temporal lobe, has long been associated with understanding words. Damage here can leave an individual fluent in speech, yet producing strings of words devoid of sense, a condition called fluent aphasia. This illustrates how comprehension depends on networks that map sounds to meaning. Modern imaging shows that comprehension involves broader circuits, including anterior temporal cortex, which helps represent word meanings, and angular gyrus, which integrates concepts across modalities. Together, these regions transform auditory patterns into ideas. Production of language involves a different but interconnected network. The inferior frontal gyrus, often called Broca's area, coordinates planning of speech. It links with motor cortex to control the movements of tongue, lips, and larynx, and with supplementary motor areas to sequence words into phrases. When damaged, speech becomes labored and halting, though comprehension may remain largely intact, a condition known as non-fluent aphasia. This dissociation between comprehension and production highlights how the brain distributes language across distinct circuits that must cooperate seamlessly. Between perception and production lies repetition, the ability to hear a word and say it back. This depends on arcuate fasciculus, a white matter tract connecting temporal and frontal language regions. Damage here can impair repetition, despite preserved comprehension and production, a condition called conduction aphasia. Such cases reveal the importance of connectivity. Language is not confined to isolated centers but flows along pathways linking them. Beyond basic words lies syntax, the grammar that organizes phrases into structured meaning. Parsing a sentence requires working memory to hold words, rules to interpret their order, and integration across clauses. Broca's area plays a central role in syntactic processing, but prefrontal and parietal circuits also contribute, maintaining phrases until they can be assembled. Syntax exemplifies the brain's ability to manipulate abstract structures, turning linear sound into hierarchical meaning. Semantics, the realm of meaning, draws upon vast networks. Words evoke not only definitions, but sensory associations, memories, and emotions. The word rose may trigger visual imagery of petals, olfactory memory of fragrance, emotional resonance of love or loss. This richness arises because language taps distributed cortical areas: visual cortex for shape, olfactory cortex for smell, limbic circuits for affect. Thus, meaning is not stored in a single place, but distributed, with language networks serving as pointers that activate relevant associations. Language is also profoundly social. Pragmatics, the use of language in context, requires theory of mind circuits to infer what others intend. A phrase like "it's cold in here" may function as observation, complaint, or request, depending on situation. Prefrontal and temporal parietal regions collaborate to interpret such cues. This capacity allows humans to coordinate, persuade, and share subtle shades of meaning, transforming language into a vehicle for culture. Developmentally, language emerges from early exposure and interaction. Infants are born with sensitivity to a wide range of phonemes, but gradually tuned to those used in their environment. Babbling evolves into words, words into sentences, through a dance of maturation and learning. Critical periods exist. Children deprived of language input during early years may never fully acquire grammar, underscoring the importance of early experience. Yet even within these constraints, the brain shows remarkable plasticity, with second languages often mastered in youth and learned, though with greater effort, in adulthood. Bilingualism reveals further insights. Brains that juggle multiple languages often show enhanced connectivity between control networks, as they must continually manage competition between vocabularies. Bilingual individuals may switch languages fluidly depending on context, an ability relying on prefrontal circuits for cognitive control. Research suggests that bilingualism can confer cognitive advantages in flexibility and delay of age-related decline. Though findings remain debated, what is clear is that the human brain is uniquely suited to handle multiple linguistic systems. Disorders of language illuminate its neural architecture. Aphasia following stroke can take many forms: non-fluent, fluent, conduction, anomic. Each reflects damage to specific nodes or connections, yet recovery is often possible through plasticity and therapy, as surviving circuits reorganize. Developmental language disorders, such as dyslexia, involve difficulties in mapping letters to sounds, often linked to atypical connectivity in temporal and parietal regions. These conditions reveal both the fragility and resilience of language networks. Language does not exist in isolation from other cognitive domains. It is deeply intertwined with memory, as words help encode and retrieve experiences. It interacts with emotion, as tone and choice of words shape feelings. It influences thought itself. Some theories argue that language structures how we categorize the world, though others emphasize that thought can exist independently. Evidence suggests a nuanced view. Language enhances many forms of cognition, but is not identical to them. Reflecting on language also raises evolutionary questions. How did such a complex system arise? Comparative studies show that other species communicate through calls and gestures, but human language is unique in its generativity, the ability to create infinite new sentences from finite rules. Fossil and genetic evidence suggest gradual evolution of vocal control, auditory specialization, and neural expansion, culminating in circuits that support syntax and semantics. Language may have co-evolved with social cooperation, as groups with better communication gain survival advantage. In daily life, language saturates experience. It frames internal monologues, guides planning, and carries memory. It allows humor, poetry, argument, and instruction. Its richness lies not only in conveying information, but in evoking imagery, shaping identity, and linking generations through shared stories. The brain's orchestration of language, from perception to production to meaning, is thus one of its most profound achievements. Imagine conversation as a river. Sounds flow into ears, are parsed into words, linked to concepts, and reshaped into replies, which then flow outward as speech. At every moment, multiple brain regions coordinate to sustain this exchange, allowing minds to connect. That such coordination feels effortless testifies to the efficiency of these networks, honed by evolution and experience. As this extended reflection draws to a close, hold the image of language not as a single faculty, but as a web stretching across the brain, binding perception, memory, emotion, and social understanding. Through it, we not only describe the world but create it, sharing inner experience with others. In the next stage of our journey, we will turn from language to music, exploring how rhythm, melody, and harmony engage the brain in ways both similar to and distinct from speech, stirring emotions and binding communities through sound. Music is as ancient as humanity, resonating across cultures and epochs, woven into ritual, celebration, and solace. It moves us in ways both universal and deeply personal, capable of evoking joy, grief, nostalgia, or transcendence. Unlike language, which conveys meaning explicitly, music often communicates without words, shaping emotion and synchronizing minds through rhythm and melody. To understand how the brain engages with music is to explore a landscape where perception, memory, movement, and feeling converge. The journey of music in the brain begins with sound. Vibrations of air are captured by the ear, transduced by the cochlea into neural impulses, and relayed to auditory cortex. Within the superior temporal gyrus, neurons respond selectively to features such as pitch, timbre, and harmony. Some populations track the fine-grained frequencies of notes, while others detect broader structures such as chords and scales. This hierarchy allows the brain to move from raw acoustics to the recognition of melody. Rhythm engages both auditory and motor circuits. Even when listening passively, motor regions such as premotor cortex and basal ganglia become active, reflecting an embodied anticipation of beats. The cerebellum contributes to precise timing, predicting the intervals between notes. This explains why listeners often tap their feet or nod their heads, even without conscious intention. Rhythm links sound to movement, a connection that may underlie music's power to unify groups in dance or marching. Melody and harmony recruit emotional circuits. The amygdala responds to dissonance with tension, while the nucleus accumbens, part of the brain's reward system, responds to consonance and resolution with pleasure. Dopamine release accompanies musical peaks, creating chills or goosebumps. These responses mirror those to food or social reward, showing that music taps fundamental circuits of motivation. Yet unlike biological rewards, music's power lies in its abstract structure, suggesting that the brain has co-opted these systems for aesthetic experience. Memory intertwines deeply with music. Songs often evoke autobiographical recall, transporting listeners to moments of childhood, romance, or loss. This effect depends on hippocampal circuits linking melodies to episodes, but also on the emotional coloring provided by amygdala and prefrontal networks. Strikingly, individuals with dementia may lose much of their declarative memory, yet retain songs, able to sing lyrics from decades past. This resilience suggests that musical memory engages widespread and redundant networks, offering a foothold for identity when other faculties fade. Language and music share overlapping substrates, yet diverge in important ways. Both rely on auditory cortex and temporal lobe structures for sound processing, and both involve syntax-like organization. Broca's area, central for language grammar, also participates in processing musical structure, such as unexpected chord progressions. Yet while language aims at denotation, music emphasizes connotation, shaping effective contours rather than explicit propositions. This difference highlights the brain's capacity to use shared circuits flexibly for different modes of expression. Cross-cultural studies reveal both universals and diversity. Rhythmic entrainment appears universal. People across cultures synchronize to beats, though preferred meters vary. Scales differ widely, yet certain intervals, such as octaves, are nearly universal, perhaps reflecting auditory system organization. Emotional responses to tempo and mode show broad commonalities. Fast major pieces tend to sound happy, slow minor pieces sad, though cultural learning shapes nuance. These patterns suggest that biology provides a foundation on which culture builds endless variety. Music also plays a powerful role in social bonding. Group singing, drumming, or chanting synchronize not only movements but physiological states, aligning heart rates and breathing. This synchronization fosters a sense of unity and cooperation, which may explain why music is central to rituals, from religious ceremonies to national anthems. Brain imaging shows increased release of oxytocin, a hormone linked to social bonding, during group music making. Thus, music may serve as a biological glue for communities. Clinical applications harness these effects. Music therapy has been used to support rehabilitation in stroke, helping patients regain speech through melodic intonation. In Parkinson's disease, rhythmic cues can improve gait, leveraging the coupling between rhythm and motor circuits. In mental health, music can reduce anxiety and depression, offering regulation of mood and a sense of connection. While mechanisms remain under study, evidence suggests that music engages broad networks, providing multiple entry points for healing. From a developmental perspective, infants are immersed in music-like interaction from birth. Caregivers use exaggerated intonation, rhythm, and melody in motherese, which captures attention and supports language learning. Infants show sensitivity to beat and contour before understanding words, suggesting that musical perception is foundational. As children grow, singing and play with rhythm foster cognitive and motor development. This early exposure may lay groundwork for lifelong responsiveness to music. Creativity in music composition and improvisation involves prefrontal circuits that balance constraint with freedom. During improvisation, activity in medial prefrontal cortex rises, while lateral prefrontal regions linked to self-monitoring decrease, suggesting a state of flow where ideas emerge with reduced inhibition. At the same time, motor and auditory regions coordinate, translating internal imagery into sound. This interplay illustrates how creativity arises from the dynamic balance between control and release. Neuroplasticity is evident in musicians' brains. Years of training reshape auditory cortex, enlarge representations of practiced tones, and strengthen connectivity between auditory and motor areas. The corpus callosum may show increased size in musicians, reflecting enhanced interhemispheric communication. These changes illustrate how sustained practice sculpts the brain, demonstrating its capacity to adapt to cultural demands. Yet even non-musicians show plasticity after short-term training, underscoring the accessibility of musical engagement. Philosophically, music raises questions about meaning. Unlike language, it lacks explicit reference, yet it conveys profound significance. Some theorists argue that music mirrors patterns of movement and emotion, resonating with embodied experience. Others suggest it exploits predictive coding. The brain anticipates sequences, and the tension between expectation and outcome produces pleasure. Whatever the mechanism, music demonstrates that meaning can emerge without words, through structure, rhythm, and resonance. In daily life, music regulates mood, enhances exercise, and marks milestones. A lullaby soothes, an anthem inspires, a requiem consoles. Its ubiquity suggests that the brain is tuned for it. Whether by adaptation or by exaptation from other functions, music transforms ordinary time into marked experience, carving memories and shaping emotion. Its effects reach from the private interior of a solitary listener to the collective energy of a concert hall. Consider the moment when a symphony swells, dissonance resolves, and an audience holds its breath before erupting in applause. In that moment, neural circuits across individuals synchronize, sharing a pulse of emotion. The brain's response to music is not confined to one listener, but echoes across many, binding them into a community of feeling. This shared resonance may be one of music's most profound gifts, the ability to align hearts and minds without a single word. As we close this extended reflection, imagine music as a bridge between brain and world, linking perception with emotion, memory with identity, and individual with group. It exemplifies the brain's capacity to find beauty in pattern and meaning in sound. In the next stage of our journey, we will move from the harmonies of music to the complexities of social cognition, exploring how the brain perceives others' minds, interprets their intentions, and builds the fabric of social life. To live as a human is to live among others. Survival, growth, and identity all unfold within social contexts. The brain is not only a machine for perceiving the world and guiding action, it is also a system tuned to perceive the minds of others, to infer intentions, to share emotions, and to build relationships. Social cognition is the architecture of connection, the set of mechanisms that transform solitary awareness into shared understanding. At the core of social cognition lies the ability to recognize faces. Humans are remarkably adept at identifying individuals, even from fleeting glimpses or subtle cues. Specialized circuits in the fusiform gyrus, particularly the fusiform face area, respond selectively to faces, distinguishing them from other objects. This specialization reflects the evolutionary importance of recognizing allies, kin, and rivals. Damage to these circuits can lead to prosopagnosia, or face blindness, in which individuals perceive features but cannot identify people. Such cases reveal how crucial face recognition is for the fabric of social life. Beyond identification, the brain interprets expressions. The superior temporal sulcus is sensitive to gaze direction, facial movement, and emotional display. It helps decode whether someone is attentive, fearful, or joyful. These signals are processed rapidly, often before conscious awareness, enabling swift responses in social interaction. For example, the sudden widening of another's eyes can alert us to danger even before we locate the threat. This efficiency reflects deep integration between perceptual systems and emotion circuits, such as the amygdala. Voice also conveys rich information. Prosody, the rhythm, pitch, and tone of speech, carries cues about emotion, intention, and social stance. Auditory cortex processes these acoustic features, while prefrontal and limbic regions interpret their meaning. A phrase spoken with warmth differs profoundly from the same words spoken with sarcasm. The ability to detect such nuances allows language to carry layers of meaning beyond literal content. A central capacity of social cognition is theory of mind, the ability to attribute beliefs, desires, and knowledge to others. This capacity emerges in early childhood, often tested by tasks that ask whether a child understands that someone can hold a false belief. Neural correlates include the temporoparietal junction, medial prefrontal cortex, and posterior superior temporal sulcus. These regions activate when we imagine another's perspective, infer motives, or predict behavior. Without this capacity, social interaction would be limited to surface cues. lacking the depth of understanding that underlies empathy and cooperation. Empathy itself involves both cognitive and affective components. Cognitive empathy relies on theory of mind circuits to model others' states, while affective empathy recruits emotion circuits to share their feelings. The anterior insula and anterior cingulate activate when we perceive another's pain, mirroring our own bodily responses. This mirroring does not mean confusion between self and other, rather it provides a vicarious window into their experience. Empathy thus binds individuals, motivating care and cooperation. Social cognition also depends on memory. The hippocampus helps encode social episodes, while prefrontal cortex maintains information about reputations, alliances, and past interactions. This memory allows nuanced judgments, whom to trust, whom to avoid, how to navigate group dynamics. Without such memory, social life would collapse into isolated encounters, stripped of history and continuity. Cultural learning further shapes social cognition. Norms, values, and roles are transmitted through observation and instruction, building layers of meaning atop biological foundations. Prefrontal and temporal regions integrate these norms, guiding behavior according to cultural expectations. For example, gaze contact may signal respect in one culture and aggression in another. The brain flexibly applies learned rules to interpret signals in context. Disorders of social cognition highlight its complexity. Autism spectrum conditions often involve difficulties in theory of mind, leading to challenges in interpreting intentions or emotions. Schizophrenia may involve altered attribution of intent, sometimes producing paranoid interpretations. Damage to prefrontal cortex can impair judgment in social situations, leading to inappropriate behavior despite intact intelligence. These conditions illustrate that social cognition is not peripheral, but central to human function. Neurochemistry also plays a role. Oxytocin, sometimes called the bonding hormone, enhances trust and empathy, though its effects are context-dependent. Dopamine modulates reward from social interaction, reinforcing cooperation. Serotonin influences mood and social confidence. These neuromodulators shape the texture of social life, turning neural circuits into lived relationships. Social cognition extends beyond dyads to groups. The brain tracks group membership, distinguishing in-groups from out-groups. Amygdala responses to unfamiliar groups may reflect vigilance, while prefrontal control can regulate prejudice. Group dynamics activate reward circuits when cooperation succeeds, but can also trigger aggression when competition arises. Thus, the same mechanisms that enable solidarity can also underlie conflict. Technology now mediates much of social interaction, raising questions about how digital communication shapes the brain. Virtual faces and voices engage many of the same circuits as live interaction, though subtle cues may be lost. The absence of physical synchrony, shared gaze, synchronized movement, may reduce bonding. Yet digital platforms also allow expanded networks, connecting minds across distance. The brain adapts to these contexts, though the long-term effects remain under study. Evolutionary perspectives suggest that social cognition co-evolved with group living. Larger group sizes correlate with larger neocortex in primates, supporting the social brain hypothesis. Managing alliances, detecting cheaters, and coordinating cooperation place demands that may have driven cognitive expansion. Human language, culture, and morality all build on these foundations, extending social cognition into abstract realms of law, art, and philosophy. Reflection on social cognition reveals its subtlety. To glance at a friend's expression and sense unspoken worry, to infer irony in a phrase, to coordinate laughter in a group, these are achievements of immense neural complexity. They occur rapidly, often below awareness, yet sustain the richness of human connection. Without them, life would be solitary. With them, it becomes shared. Consider a conversation. Faces are recognized, voices parsed, emotions inferred, beliefs attributed, responses shaped. Each moment involves multiple brain regions, from fusiform gyrus to prefrontal cortex, from amygdala to temporoparietal junction. The ease with which this unfolds conceals the orchestration beneath. It is as if a vast ensemble plays in unison, producing the symphony of social life. Social cognition also involves moral judgment. When evaluating fairness, harm, or intention, regions such as ventromedial prefrontal cortex and posterior cingulate engage. These networks integrate emotion, memory, and reasoning, producing judgments that guide behavior. Moral cognition thus emerges from the same circuits that support empathy and perspective-taking, reflecting the brain's role in sustaining cooperative societies. Training and experience can refine social cognition. Practices such as mindfulness or compassion meditation enhance empathy and theory of mind, reflected in altered activity of prefrontal and insular regions. Cultural exposure broadens interpretation of cues, fostering cross-cultural competence. Even simple exercises such as storytelling or role-play strengthen the ability to imagine perspectives. These findings suggest that social cognition is not fixed but plastic, shaped across the lifespan. As we close this reflection, imagine social cognition as a mirror and a bridge. It mirrors other states within our own brain, allowing resonance. It bridges between individuals, creating shared meaning. It is the foundation of family, friendship, and community. In the next stage of our journey, we will explore how networks of regions across the brain cooperate more broadly, forming large-scale systems that sustain not only social interaction, but all aspects of cognition, binding perception, memory, and action into a coherent whole. The brain is not a collection of isolated islands, but a vast archipelago joined by currents of connectivity. Each region has local specializations, yet cognition arises from their cooperation, woven into large-scale networks. These networks are not fixed structures but dynamic configurations, flexibly engaging and disengaging depending on task, mood, and state of consciousness. To understand them is to see how the brain integrates perception, memory, attention, and emotion into the seamless flow of thought. One of the most studied networks is the default mode network. It includes medial prefrontal cortex, posterior cingulate cortex, and angular gyrus, among other areas. This network is most active when the mind is at rest, drifting through memories, imagining futures, or reflecting on self. It dims when external tasks demand focus, yet reactivates when attention turns inward. The default mode illustrates how the brain is never truly idle. Even in stillness, it generates narratives, scenarios, and a sense of continuity. In contrast, the dorsal attention network orients focus toward the external world. Anchored in intraparietal sulcus and frontal eye fields, it guides the spotlight of attention toward relevant stimuli. Whether tracking a moving object or scanning a crowded scene, this network enhances sensory processing where it is directed. Its activity often opposes that of the default mode. When one rises, the other falls, reflecting the brain's balance between outward engagement and inward thought. The salience network, centered in anterior insula and anterior cingulate, monitors the environment for important events. It evaluates signals for novelty, emotional relevance, or potential threat, and shifts resources accordingly. For example, when a sudden sound interrupts concentration, the salience network rapidly engages, recruiting attention to evaluate significance. It acts as a switchboard, coordinating transitions between default and attention networks. The executive control network, involving dorsolateral prefrontal cortex and posterior parietal cortex, maintains goals and rules. It enables planning, problem-solving, and flexible adjustment. This network interacts closely with memory systems, drawing on stored knowledge to guide behavior. When solving a puzzle, it holds the steps in working memory while suppressing distractions. The executive network demonstrates the brain's capacity for deliberate, structured thought. These networks are not confined to specific tasks. Rather, they interact constantly. Functional connectivity studies reveal that even at rest, regions show correlated activity, suggesting intrinsic organization. These patterns may provide a scaffold upon which task-specific configurations build. Variations in connectivity correlate with personality traits, cognitive abilities, and vulnerability to disorders, highlighting how networks shape individuality. Memory depends on network cooperation. The hippocampus encodes episodes, but retrieval involves interaction with prefrontal cortex, default mode regions, and sensory areas. When recalling a scene, visual cortex reactivates patterns similar to those during perception, guided by hippocampal prefrontal dialogue. This reactivation illustrates how networks recreate experience, blending fragments into coherent recollection. Emotion likewise recruits networks. The amygdala interacts with salience and executive systems, prioritizing processing of threat or reward. Connections with prefrontal regions enable regulation, dampening fear or amplifying positive affect. Dysregulation of these interactions underlies conditions such as anxiety or depression. Thus, emotional life is not the product of one structure, but of network dynamics spanning limbic and cortical regions. Consciousness itself may emerge from network integration. Some theories emphasize the global workspace, in which information becomes conscious when widely broadcast across networks. Others focus on integration of information across distributed nodes. Whatever the model, Evidence suggests that consciousness depends not on isolated activity, but on coordinated communication across regions. Anesthetics, for instance, disrupt connectivity, fragmenting networks and reducing awareness. Development shapes networks through pruning and strengthening of connections. Infants display diffuse connectivity, which becomes more specialized with experience. Adolescence brings further refinement, particularly in prefrontal circuits, supporting maturation of control and social cognition. Lifespan changes continue, with aging altering network balance, sometimes leading to declines but also to compensatory reorganization. These shifts reveal the plasticity of networks across time. Disorders often reflect network dysfunction. In schizophrenia, abnormal connectivity among default, salience, and executive networks contributes to disorganized thought and impaired reality testing. In depression, hyperconnectivity within default mode may trap individuals in rumination. In dementia, degradation of network hubs such as posterior cingulate undermines memory and orientation. These patterns show that mental illness often arises not from single lesions, but from disrupted coordination. Technological advances reveal networks with increasing precision. Functional MRI maps correlations in activity, while diffusion imaging traces structural pathways. Electrophysiology shows temporal dynamics, capturing millisecond synchrony. Together, these methods uncover not static wiring diagrams but living communication. Networks emerge, dissolve, and reconfigure within seconds, reflecting the fluid nature of cognition. The metaphor of orchestra captures their essence. Each instrument, like each region, has its timbre and range. But music arises only when they play together, guided by shifting conductors, sometimes executive control, sometimes salience, sometimes default narrative. Harmony and dissonance alternate, producing the symphony of thought. Network science provides tools to analyze these patterns, treating brain regions as nodes and connections as edges. Measures such as centrality identify hubs crucial for integration. Small World Organization describes how networks balance efficiency and resilience. These principles echo across other complex systems, from social networks to ecosystems, suggesting general laws of organization. Training and experience reshape networks. Meditation enhances connectivity between executive and default regions, supporting regulation of attention and emotion. Learning new skills strengthens task-related networks, whether motor or cognitive. Even brief interventions can alter connectivity, showing the responsiveness of networks to practice. This plasticity offers hope for rehabilitation and growth. Reflection on networks challenges reductionist views. No single area is memory, emotion, or self. Instead, these emerge from patterns of interaction. The brain is both modular and integrated, combining specialization with cooperation. This architecture enables flexibility, allowing adaptation to changing demands. It also explains why damage to one region can sometimes be compensated by reorganization across networks. Daily experience illustrates network dynamics. When daydreaming, default mode drifts through scenarios. When a task demands focus, executive and attention networks rise, suppressing distraction. When a sudden event occurs, salience network interrupts, redirecting resources. This constant choreography underlies the fluidity of thought, shifting seamlessly from inward reflection to outward action. As we consider networks, we glimpse the essence of brain function, not isolated structures but living patterns of communication. They reveal how the brain binds perception, memory, attention, and emotion into the unity of consciousness. They also remind us that individuality arises from these patterns. Each brain has its own signature of connectivity, shaped by genes, experience, and culture. As this reflection closes, imagine networks as constellations in the night sky, their stars joined by invisible lines of connection. They shift as we watch, reconfiguring into new shapes, yet sustaining coherence. In the next stage of our journey, we will turn to experimentation, asking how one might design studies to probe these patterns, and how scientific inquiry itself becomes a dialogue between question and brain. Designing an experiment to study the brain is both a technical challenge and a philosophical exercise. It requires translating a question about mind into a procedure that can generate observable data, and then interpreting those data in ways that illuminate hidden processes. Unlike inanimate systems, the brain cannot be opened like a clock to reveal fixed gears. Its workings must be inferred indirectly, from patterns of behavior, neural activity, and the interplay between them. To reflect on how one might design such studies is to step into the heart of cognitive neuroscience. The first step in any experiment is formulating the question. Good questions are precise, yet meaningful, small enough to be answerable yet large enough to matter. For instance, rather than asking, "How does memory work?" one might ask, "Do people retrieve visual details of scenes from the same cortical regions engaged when they first perceived them?" This question is specific, testable, and connects behavior to neural representation. The art of questioning lies in narrowing the focus without losing sight of the broader significance. Once a question is posed, hypotheses take form. These are not mere guesses but structured predictions grounded in theory. A researcher might hypothesize that reactivation of sensory regions during recall reflects the mechanism by which memory reconstructs experience. Another hypothesis might propose that retrieval engages distinct circuits, independent of initial perception. Competing hypotheses sharpen experimental design, ensuring that results can distinguish between alternatives rather than simply confirming intuition. Operationalization follows. Abstract concepts must be translated into measurable variables. Memory, for example, becomes performance on recognition tests. Attention becomes reaction time to stimuli. Emotion becomes self-reported ratings or physiological signals such as heart rate. In neuroscience, operationalization extends to neural measures. Activity in a region is measured by blood flow, oscillatory patterns recorded from scalp or spike trains from electrodes. Each measure is partial, reflecting one aspect of the underlying process. Choosing methods is the next critical step. Functional MRI offers spatial resolution, showing which regions are active, but lags in time. EEG and MEG offer millisecond precision but blur spatial localization. Lesion studies reveal necessity, but lack experimental control. Animal physiology allows invasive recording, but may not generalize fully to humans. Often, the strongest experiments combine methods, triangulating evidence from different perspectives. The choice of method reflects the trade-off between spatial detail, temporal resolution, and invasiveness. Designing stimuli and tasks requires creativity. Stimuli must be controlled to isolate variables while remaining ecologically valid. A study of face perception might use standardized photographs to control expression and lighting, yet must also capture the variability of real encounters. Tasks must engage the process of interest without introducing confounds. For example, testing memory for words requires ensuring that differences in familiarity or emotional content do not bias results. Each choice shapes what the experiment can reveal. Control conditions are essential. To claim that a region supports face perception, one must compare responses to faces versus other objects matched for complexity. To show that memory reactivation occurs, one must compare recall to mere imagination. Controls rule out alternative explanations, isolating the factor of interest. Without controls, results remain ambiguous, open to interpretation. Participants bring variability. Age, culture, language, and individual differences shape neural responses. Decisions must be made about sample size, inclusion criteria, and balancing groups. Too small a sample risks spurious findings. Too narrow a sample risks limited generality. Ethical considerations also guide recruitment, ensuring respect, consent, and protection. The brain is not only an object of study but the seat of personhood. Experiments must honor that dignity. Data collection requires rigor. In imaging, movement artifacts must be minimized. In electrophysiology, noise must be filtered. Trials must be sufficient to yield reliable averages, yet short enough to avoid fatigue. Procedures must be standardized, ensuring comparability across participants. The researcher's attention to detail shapes the clarity of signal against background noise. Analysis transforms raw data into patterns. Statistical methods test whether observed differences are reliable or due to chance. Correlations reveal associations. Regressions model predictors. Multivariate analyses capture distributed patterns. In neuroimaging, machine learning can decode neural signatures of perception or thought. Yet analysis is not mechanical. It requires interpretation, linking numbers back to theory. Misapplied statistics can mislead, while careful analysis illuminates hidden dynamics. Interpretation demands humility. A region active during a task does not mean it is dedicated solely to that function. Correlation does not prove causation. Null results do not always indicate absence of effect, but may reflect inadequate sensitivity. Theories must accommodate complexity and uncertainty. The most robust findings emerge from converging evidence across studies and methods. Replication strengthens conclusions. A single experiment provides a glimpse, but repeated studies across labs establish reliability. The replication crisis in psychology and neuroscience reminds us of the importance of transparency, pre-registration, and open data. Science advances not by isolated discoveries but by cumulative verification. Each experiment becomes part of a larger conversation, its value measured by how it integrates with others. Ethical reflection permeates experimental design. Invasive studies raise questions of risk, imaging studies raise issues of privacy, as patterns of activity may reveal personal traits. Informed consent, confidentiality, and beneficence guide practice. Researchers must weigh potential benefits of knowledge against risks to participants. This responsibility is heightened when studying vulnerable populations, such as children or patients with disorders. Experimentation also reflects cultural and philosophical assumptions. Questions deemed important often mirror societal values. In the past, emphasis on deficits overshadowed strengths. Today, interest grows in resilience, creativity, and well-being. Awareness of bias ensures that research serves diverse populations and addresses broad human concerns. The brain is universal, but how we study it reflects our perspectives. Emerging technologies expand possibilities. Optogenetics allows precise control of neural circuits in animals, illuminating causal mechanisms. Non-invasive stimulation, such as transcranial magnetic or electrical stimulation, offers tools to modulate human brain activity temporarily. Computational modeling simulates networks, generating predictions for experiments. Each advance opens new questions, demanding careful design to harness potential without overreach. At its heart, experimentation is a dialogue between theory and evidence. Theory suggests predictions, experiments test them, results refine theory. This cycle drives progress, gradually building models of how the brain creates mind. Yet no experiment provides final truth. Each is a step, partial and provisional, in the ongoing quest. For students and researchers, designing an experiment is an act of imagination grounded in rigor. It requires curiosity to pose questions, creativity to design tasks, discipline to control variables, and humility to interpret results. It is both science and art, a structured yet open-ended pursuit. Reflecting on experimental design also highlights its human dimension. Behind every data point lies a participant who offered time and trust, and a researcher who invested effort and care. The enterprise is collaborative, linking individuals across roles and generations. Each experiment becomes a thread in the tapestry of knowledge, woven together by collective endeavor. Daily life itself can be seen as informal experimentation. We test strategies for memory, adjust attention, regulate emotion, and observe outcomes. The scientific method formalizes this process, adding control, measurement, and replication. Yet at root, both rest on the same curiosity, the desire to understand how experience arises. As we close this reflection, imagine the laboratory as a place where questions meet methods, where minds probe their own machinery. Designing an experiment is not only about producing results, but about cultivating inquiry, shaping how we think about thinking. In the next stage of our journey, we will draw the threads together, considering how all we have explored from neurons to networks, from perception to social life, culminates in the integrated experience of being human. To bring together the many threads of our exploration is to step back and view the human brain, not as a collection of isolated topics, language here, music there, memory in one corner, networks in another, but as a living whole. The brain is a system of systems, a layered architecture where biology meets experience, where ancient circuits interweave with new adaptations, where individual uniqueness emerges from universal structures. In reflecting on this integrated view, we can see how perception, memory, emotion, and social connection converge to form the flow of daily life. At the most fundamental level, the brain is built from neurons and glia, cells that communicate through electrical and chemical signals. These elements, though simple in isolation, give rise to astonishing complexity when arranged into circuits and networks. Action potentials propagate down axons, neurotransmitters diffuse across synapses, and receptor cascades trigger responses. Out of these micro-level processes arise the capacities we experience as thought, feeling, and awareness. The mystery lies not in the existence of neurons, but in how their collective activity scales into the richness of mind. Structure provides the scaffolding. The brainstem regulates breathing, heart rate, and arousal, anchoring life itself. The cerebellum refines movement and timing, integrating with both motor and cognitive functions. The limbic system supports emotion and memory, linking bodily states with experience. The neocortex, expanded in humans, enables perception, language, reasoning, and planning. Each region has its specialization, yet none acts alone. Fibers weave them into webs of communication, ensuring that no perception remains untouched by memory, no action uncolored by emotion. Perception demonstrates this integration. When light strikes the retina, signals travel to visual cortex, but perception of a scene is not confined there. Memory enriches it with familiarity, emotion colors it with significance, and attention selects what to emphasize. The same applies to sound, touch, and smell. Sensory input is never raw, but always interpreted through layers of context. The brain constructs the world we experience, filtering and shaping it through its networks. Memory reveals how the past infuses the present. The hippocampus encodes episodes, yet retrieval involves distributed reactivation across sensory and associative regions. A smell may trigger childhood recollection. A song may transport us to adolescence. These memories are not stored as perfect records but reconstructed, blending fragments with current perspective. The result is a narrative self, a sense of continuity across time. Without memory, identity fractures. With it, we carry our past into every moment. Emotion adds intensity and meaning. Circuits involving the amygdala, insula, and prefrontal cortex evaluate stimuli for significance. They influence attention, prioritize memory, and shape decision-making. Fear alerts us to danger. Joy binds us to others. Sadness signals loss. Emotions are not interruptions of cognition but essential components, guiding adaptive responses. Their regulation depends on networks that balance reactivity with control, allowing us to respond flexibly rather than reflexively. Language transforms cognition into communication. By linking sounds or symbols to concepts, it allows sharing of thought across individuals. Neural circuits for production, comprehension, and syntax collaborate to produce fluent expression. Language interweaves with memory, emotion, and social cognition, enabling storytelling, persuasion, and teaching. It structures thought, offering categories and metaphors that shape how we perceive the world. Without it, minds would remain largely private. With it, they join into cultures. Music offers another window into integration. It engages auditory cortex, motor circuits, emotion networks, and memory systems. Its rhythms synchronize bodies. Its melodies evoke feelings. Its harmonies mirror social cooperation. Unlike language, music often communicates without explicit reference, yet it carries profound meaning. It illustrates how the brain finds beauty in pattern, deriving reward from prediction and resolution. Music exemplifies how cognition is not only about utility, but also about aesthetic experience. Social cognition binds individuals into groups. Recognition of faces, interpretation of expressions, attribution of beliefs, all these depend on specialized circuits. Empathy allows resonance with others' states, while theory of mind enables understanding of perspectives. Social cognition is shaped by memory, emotion, and culture, integrating multiple domains into the subtle art of interaction. It sustains families, friendships, and societies, making possible cooperation on scales far beyond kinship. Networks provide the infrastructure for integration. Default mode, attention, salience, and executive networks interact dynamically, supporting shifts between inward reflection and outward action. Connectivity allows information to flow across domains, binding perception, memory, emotion, and social cognition into unified experience. The orchestration of networks is the basis of consciousness itself, enabling a coherent stream from diverse neural activity. Consciousness remains one of the deepest puzzles. We know it depends on integrated activity, yet how subjective experience arises from physical processes is still unclear. Some theories propose global broadcasting of information, others emphasize integration and differentiation. Whatever the mechanism, consciousness reflects the coordination of networks across scales, from local circuits to distributed systems. It is the ground of experience, the canvas upon which perception, memory, and thought are painted. The brain is plastic, shaped by experience. Learning rewires circuits, practice strengthens connections, and culture imprints patterns. Musicians, bilinguals, meditators, all show changes in connectivity and function. Even injury does not fix destiny. Surviving circuits can reorganize, supporting recovery. Plasticity demonstrates that the brain is not static hardware but living tissue that grows with use. It is both resilient and vulnerable, shaped continuously across the lifespan. Disorders remind us of this vulnerability. Stroke, trauma, degeneration, and developmental conditions disrupt networks, producing deficits in language, memory, emotion, or social cognition. Yet studying these disruptions illuminates normal function, revealing what each circuit contributes. It also underscores the importance of clinical care and rehabilitation, translating neuroscience into practical support. The fragility of the brain deepens appreciation for its resilience. Culturally, the brain reflects and sustains human diversity. Different languages, musical traditions, and social norms sculpt neural circuits in unique ways. Yet beneath these variations lies a shared architecture, enabling cross-cultural understanding. The brain is at once universal and particular, carrying the imprint of both evolution and environment. This duality allows humanity to share common ground while flourishing in variety. Philosophical reflection arises naturally. The brain is the physical organ of mind, yet subjective experience feels more than material. How does matter produce meaning? How do neurons give rise to love, art, or morality? These questions may never be answered fully by science alone, yet neuroscience provides insights that ground them. It shows that mind is embodied, that self arises from networked activity, that free will may coexist with constraints of circuitry. The mystery is not diminished by understanding, rather it deepens with appreciation. Daily life illustrates the integrated brain. A morning walk engages sensory perception, motor coordination, memory of routes, and reflection on plans. An encounter with a friend activates face recognition, empathy, language, and shared memories. Listening to music while cooking blends auditory pleasure with motor routines and emotional regulation. Each simple activity reflects a tapestry of circuits and networks, interwoven seamlessly. The ordinariness of these moments belies their complexity. The integrated view also reveals how science itself is a collective cognition. Experiments are designed, data collected, results shared, theories debated. The process mirrors the brain's networks, individual researchers as nodes, journals as connections, conferences as hubs. Knowledge grows through communication, much as consciousness arises from integrated activity. Neuroscience not only studies networks but enacts them, building understanding through collaboration. As we draw our journey toward its close, consider the brain as both a biological organ and a philosophical mirror. It is tissue, blood, and electricity, yet also thought, memory, and identity. It grounds us in survival while lifting us into art and morality. It is the most intimate part of us, yet also the most mysterious. Studying it illuminates who we are, yet leaves us humbled by what remains unknown. Integration is the theme that binds everything together. Neurons integrate signals, regions integrate functions, networks integrate domains, and consciousness integrates experience. This integration allows the brain to be both specialized and flexible, both stable and adaptive. It allows us to perceive a coherent world, to remember a continuous self, to feel emotions, to speak, to sing, to love, to imagine. Integration is the essence of mind. As this reflection concludes, imagine the brain as a vast landscape where rivers of connectivity flow between mountains of specialization, valleys of memory, forests of emotion, and plains of perception. This landscape is alive, changing with every experience, yet continuous across a lifetime. It is at once deeply personal and profoundly universal. In recognizing its complexity and integration, we glimpse not only the organ of thought, but the foundation of humanity itself.